on this thing because it doesn't happen so far. It used to be you really were expected by your jacket. Before us, when Max Down was around, which is just kind of ridiculous because, it's like...
Okay, good afternoon, everybody. We would like to start. So, Professor Dani Leviathan, Director of Tel Aviv University, Professor Ehud Bazit, the Vice President for Research and Development, Professor David Baker, Professor Martin Grubeler, and Professor Jonathan Weissman, the laureate of the 2008 Sackler Prize, dear colleagues and friends. In the name of the prize committee, Professor Kozlov and I would like to welcome you all to the ceremony of the Raymond and Beverly Sackler Prize in Biophysics. In this third year, the Tel Aviv University is awarding an international prize in biophysics. This is the third year that Tel Aviv University is awarding the International Prize in Biophysics. The prize has been established through the generosity of Raymond and Beverly Sackler, who wish to promote excellence in this emerging and interdisciplinary field of science, a field that lies in the crossroad between physics, biology, chemistry, medical sciences, and engineering. The prize is awarded each year in a different area of biophysics. This year, the area has been chosen to be physics of structure formation and self-assembly of protein and nucleic acids. More specifically, this field relates to investigation of folding, regulation, and conformation of proteins and other biological macromolecules. The disciplines involved range from bioinformatics and computational biophysics through experimental physics and modeling, all the way to cell and molecular biology. For the year 2008, the prize committee has decided to award the Sackler Prize to 3 internationally distinguished scientists with an extraordinary track record of biophysical research. I call upon Professor Dani Leviathan, Director of Tel Aviv University, to welcome the laureates.
Thank you, David. It's always a pleasure to come to my home faculty, the Faculty of Exact Sciences, to see the faces. We don't have many opportunities, and always the presentation of this prize and prizes like this bring me over, and that's very nice. I was very impressed by the little I read, of course, in these small things about the achievements of the laureates, and it is indeed a very important subject that is developing, interdisciplinary subject. We see here people from the school, from the Faculty of Medicine, people from the Faculty of Life Sciences, and of course from the Faculty of Exact Sciences. And it's really important to us to mention again, although David mentioned it already and we mentioned it the third time, because it's the third time we are giving the prize, that we really are grateful to Dr. Raymond Sackler for this prize and his other contributions and help that we get at Tel Aviv University due to his generosity. I'm sure that the 3 new laureates are going to carry on with pride. Having received this prize, they will be in a long line, or not so long because it started only 2 years ago, but eventually they will be in a long line of laureates in a prestigious prize. And I want to thank both the committee and especially David Endelman and Mikhail Kozlov for handling the prize for the third time, and you will continue. Thank you.
So I would like to call now upon Professor Ehud Gazit, the Vice President of Research and Development, to welcome the laureate.
Thank you, David. It's really a pleasure to welcome you here and to congratulate you on this award. For me, this award is really related to my own work, self-assembly of proteins. So I really feel at home, and I really could appreciate the high-level, scientific level, as well as personal level of the laureates. As was mentioned by the rector, a tradition of an award is being is the product of the level of awardees, and in this case, this is a young award. It's not very highly in its prize, although it's significant. But as many other prizes, we do hope that it will be a predictor for many more prizes in the future. And I think we have excellent young investigators who are really in the top of their field. This award that was is supported by Dr. Sackler is very important. I think that the field of biophysics is one of the fields that will dominate the scientific world in the 21st century. And Tel Aviv University have very strong background both in physics or many forms of physics as well as biology. And this combination and the interaction of our faculty members together with this excellent awardees of this Sackler Prize is very important. And Tel Aviv University is committed to research, excellent research. We are the largest university in Israel, the most diverse one, and by awarding this award in biophysics, this is our commitment with the generous support of Dr. Sackler to support this field. So, congratulations again. Welcome to Tel Aviv University. We are very happy that you joined the family, young family, but a very distinguished one of the awardees of this award. Thank you.
I call now upon Professor Michael Kozlov to introduce the prize to the first laureate, Professor David Baker.
So, shalom to everyone. It's my great pleasure and honor to introduce Professor David Baker, the recipient of the 2008 International Sackler Prize in Biophysics. I must say that David had a very long and hard trip this time to Israel. This trip took probably more than 24 hours, I would expect. He landed today at 8 o'clock, and we are afraid he will not be able to reach the ceremony, and we are twice as happy to have David with us. He made it, and thank you very much for this effort. So David received his undergraduate degree from Harvard University and his PhD degree from the University of California at Berkeley. After a postdoctoral period at University of California, San Francisco, he joined the Department of Biochemistry at the University of Washington, and this was in 1993. And since then, he has been at the University of Seattle, University of Washington in Seattle. And now, currently, he is a full professor and Howard Hughes investigator at the University of Washington in Seattle. Few works about David's achievements, at least a part of David's achievements. The field David has been working in can be related to computational research of protein folding and protein structure. David developed techniques to use short sequence fragments to infer structural motif for protein segments. He invented an optimization procedure called the Rosetta procedure, which pieces together protein fragments into a three-dimensional structure which approximates the real protein structure. His fundamental achievements include many things, among them an improved potential function for protein interaction, relationship between the rate of protein folding and protein structure, accurate design, a design procedure for protein-protein docking, and design of new enzyme activities. The highly sophisticated computational tools and results developed by David have been successfully combined with experimental data coming from NMR and X-ray studies. The prize is awarded to Professor David Becker for his seminal contribution to computational studies of protein folding, structure, and reactions. I call upon Professor Dani Leviathan, Director of the University, to award the prize to Professor Becker.
So it's my great pleasure and privilege to introduce the Professor Martin Grubele, the second laureate, the second Sackler laureate for 2008. Martin did both his undergraduate and graduate studies at the University of California, Berkeley, completing his PhD in 1988. After a postdoctoral fellowship at Caltech, Martin joined in 1992 the University of Illinois at Urbana-Champaign, where he is currently the James Eisner Professor of Chemistry, Physics, Biophysics, and Computational Biology, all four. Martin studied very fast processes which last less than a millisecond and that mediate the structural dynamics of complex biomolecules and in particular protein which undergo folding. His work includes computational and spectroscopy studies of energy flow within molecules and the role played by solvation dynamics in protein folding. One of Martin's methodological achievements is a laser temperature jump technique used to study refolding of protein. Martin was the first to resolve nanosecond and microsecond events of protein refolding to its native state. The Sackler Prize is awarded to Professor Martin Grubele for his seminal contribution to experimental studies of fast processes mediating the structural dynamics of complex biomolecular systems. I call upon again the rector, Professor Dani Leviathan, to award the prize to Professor Grubele. I call now upon Professor Ehud Gazit, the Vice President, to introduce Professor Jonathan Weissman, the third laureate.
Thank you. So it's really a great pleasure and honor to introduce Professor Jonathan Weissman, the recipient of the 2008 Sackler Prize in Biophysics. I know Jonathan already from the previous century when both of us were young. Actually, I worked for him as a reviewer in the Journal of Molecular Biology, where he's an editor. And the first thing that I said when we met is my apologies for being late for my last review. But other than that, Jonathan is really a superb scholar. He completed his undergraduate studies at Harvard University, received his PhD in physics from MIT in 1993. After his postdoctoral work at Yale University, he joined the University of California in San Francisco in 1996, where he's now a full professor of cellular and molecular pharmacology. and of biochemistry and biophysics. He's also the Howard Hughes Investigator, a very prestigious appointment. Jonathan's major achievement is the creation of synthetic prion forms of the yeast sub-35 protein and the introduction back to yeast. His experiment with yeast fulfilled the key prediction of the protein-only hypothesis of prion inheritance, a very important discovery from the medical as well as basic science point of view. Weissman's work resolved the nature of prion strain behavior, his development of techniques for transferring in vitro converted material into intact yeast, enabled biophysical characterization of strain difference by EPR and NMR techniques. This prize is awarded to Jonathan Weissman for his seminal contribution to the study of protein folding and the formation and transition of prions. I call upon the rector to award the prize to Professor Weissman.
So this concludes the formal part of the Sackler Prize Award. We will take a short break for about in 20 minutes and return at 14:45 for the scientific part, where the three laureates will give each 45 minutes talk. You have the abstract of the talks. And I would like to end by thanking Miss Nina Elazar-Wolf, who just stood up. She was the Prize Committee Administrator. She helped us tremendously over the last month, taking care of all the fine details and ensuring that this event will be executed in the most flawless and smooth way. So Nina, toda raba. I think that the three laureates will also like to make very small speeches. So if you, okay, so maybe we start with David. Would you like to start?
Well, I'm very honored to be here as a Sackler awardee. I think it's a great honor, and I want to thank a lot of people who put a lot of hard work into this. Professor Andelman and Kozlov, and I want to thank my parents for making it all the way out here with me. It did take us at least 24 hours. I want to thank Nina for her constant attention, including many phone calls in various places along the way, and also for, I just should tell you that when you look at me, you're really looking more at Nina's husband than me. And so I want to thank her very much for all of her help, and thank all the organizers for putting this on. Thank you.
Martin, would you like to say so?
It was my pleasure to actually give a lecture on Friday in the Sackler Auditorium in the Sackler Building at Columbia University in the States. So the same family, and it's actually wonderful that they have undertaken to support fundamental research in this way. And so with that, and not much further ado, the only thing I want to say is, if you ever for some reason want to move to Champaign-Urbana, let me know and we'll hire you.
Okay, last but not least, Jonathan.
Thank you. First, I'd like to thank the Sackler Committee, David and Michael, and everyone else for the honor. It's a wonderful honor to be here. And also, of course, the Sacklers for supporting this research prize and for all their support in basic science. So as someone who was trained once upon a time in physics and has moved off into the largely inexact sciences, it's a particular honor to be back here and to be awarded for a biophysics prize. And also, of course, thank Nina, who's chaperoned us, all three of us, through this, and we really wouldn't be here without her. And then just thank, of course, my family for putting up with all the work, and of course also all the people in my lab who really I'm just representing because it's their work. Thanks.
So this concludes this part of the ceremony, and we will be back in 15 minutes.
It's a special pleasure for me to open the session, both as the Dean of the Faculty of Exact Sciences, and especially as a structured mathematician. So the name spoken in the document does not necessarily sound familiar to cities, although I'm not entirely sure that they are not. So since all of our speakers have been already presented in the previous session, I'll take the liberty not to present them and give them additional 3 minutes to talk. So without further introduction, I will call Professor David Baker from the University of Washington, Seattle to give a talk on the prediction of structure to design of function.
Well, thank you very much. And of course, I want to thank the Sacklers for making all of this possible. So I'm going to talk today about the prediction of structure and the design of functions. So in nature, we have very large numbers of sequences of biological macromolecules. These sequences, of course, are being determined at a very rapid pace with the sequencing of many of the genomes of many organisms. These sequences encode protein molecules and RNA molecules and other biomolecules which adopt specific structures and which allow them to carry out specific functions and have very specific interactions. And this is really what life at the molecular level depends upon, the precise interactions of these marvelous macromolecules that are encoded by genome sequences. So the first problem then is, how does one go from linear sequences to three-dimensional structures and interactions and functions? This is a prediction problem to predict to predict the structures and functions of proteins from their sequences. Now, while in nature we have a very large number of fascinating molecules which do marvelous things, of course there's an even larger set of macromolecules which nature didn't sample during evolution, either because those macromolecules weren't useful, or simply because the evolutionary process never hit upon them. So one can imagine a whole world of new biomolecules which were never sampled during evolution, and that brings up the second problem, which is the design problem. How do we go from new functions, new interactions, new catalysts, say, backwards to identify the sequences which will encode those? So this is the prediction problem, forward problem, going from sequences to structures and functions, and this is the inverse problem, the design problem, going from structures and functions back to sequences. These are the two problems that my research group works on that I'll give you an overview of today. The way that we approach these problems is to develop a model of the energetics of the interactions within and between macromolecules. Given such a model, the prediction problem is simply, given the sequence of, for example, of a protein, find the lowest energy structure. So here we keep the sequence constant and search for the lowest energy structure. The inverse problem, the design problem, is, given the structure or a function we want, find the lowest energy sequence. So in both cases, we're making use of this model of the energetics. It's just that in one case, we are starting with naturally occurring sequences, searching for the lowest energy structure, whereas in the design case, we're starting with the structure that doesn't exist, or a function that doesn't exist, and trying to find the lowest energy sequence that adopts that structure or has that function. So we do this in the context of a program we've been developing over the years called Rosetta, and an important aspect of these two problems, which I want to emphasize, is that they both are hard problems where one has plenty of opportunities to fail. So in prediction problems, one very often predicts the structure of a protein and gets it and finds the lowest energy state, which is actually very, very different from the experimentally observed state. And this gives us feedback to improve this model. Likewise, in the design case, we find a sequence that is predicted as a very low energy in a particular structure. We make the sequence and find experimentally doesn't adopt that structure. This gives us feedback to improve the model. So as we've pursued these prediction and design problems, we've gotten lots of feedback to improve the underlying model. And I think this is a very important part of developing any kind of computational model in biology or in anything else, that one needs very stringent tests to determine whether one's on the right track and how to improve the model. Okay, so I'm going to begin with the first problem, the prediction problem. This is a movie showing a protein holding up from an extended chain. One of the problems one's faced in the prediction challenges is the very, very large space of possible conformations that can be adopted by a polypeptide chain. Because of this, one needs to search the space very, very broadly. And so in the first phase of the process, we carry out a very coarse-grained search where we integrate out a lot of the degrees of freedom, all the side chains, for example. And in this very coarse-grained model, where the primary contributions which are being minimized are hydrophobic interactions. So there's a drive to bury the hydrophobic residues, make hydrogen bonds between the backbone and so forth. So this process, because we've smoothed out the energy so significantly and integrated out so many degrees of freedom, is very rapid. The calculation doesn't take much longer than what you see here on the computer in front of you, but we can't calculate energies very accurately because we've integrated out so many of the key degrees of freedom. So in the next step, we take the lowest energy structure found in that initial coarse-grained search, and now we add in all the atomic detail. You can see it here, all of the side chains, and we model the interactions at this stage as realistically as possible. So we treat hydrogen bonding explicitly. We have van der Waals interactions between the nonpolar atoms here. We model the solvation effects on the polar interactions. And so while we can calculate energies more accurately, it takes much longer to do the sampling. And you can see during this calculation that the backbone of this protein is not moving very far. So one sort of picture I'll be showing a number of times during this talk is this one here. On this axis I have the energy, or free energy. On this axis I have the distance from a reference structure, in this case the actual experimentally determined structure. And each point here represents the lowest energy structure sampled on that second high-resolution search. So we carry out, for each of these points, we've carried out an independent low-resolution search. That was that first movie. Then the high-resolution search, and we take the lowest energy point, and we record its energy. That's on this axis, and its distance from the experimentally determined structure. And so what you can see here are two things. First of all, that different trajectories are ending up in very different places on this landscape, so with very different energies and very different distances from the correct structure. This is an illustration of a very large number of local minima on this landscape. A second thing that you can observe is that if you start these points here, instead of starting with an extended chain, we're starting with the experimentally determined structure, and you can see that one samples much lower energies than one does starting from an extended chain, and these structures also stay quite close to the native structure. So if you start out in the native free energy minimum, by and large you stay there, and by and large the structures that one samples are lower in energy than those one samples from an extended chain. Now a third thing is that in this case you can see there were a couple of structures, a couple of the trajectories where we're starting from an extended chain where which actually sampled
close enough to this native free energy basin for the free energy to drop a bit. And in such cases, one can pick out from this very large population of structures these ones based on their lower free energy. And in such cases, one sees that these low energy structures are very, very similar to the actual experimentally determined structures. So the experimentally determined structure here is in red. The lowest energy structure sampled in all these trajectories is in blue. And what you can see is that they're quite superimposable. In particular, in the core, these side chains here are packing together in the jigsaw puzzle-like way that is responsible for that dramatic drop in energy close to the native structure. So we've seen this phenomenon we've observed over and over in many different types of scenarios. So here, certain types of proteins, as you know, form symmetric homodimers. So in this case, we're carrying out the low-resolution search, enforcing symmetry between the two chains. And in the second stage, we then go to a high-resolution model, very much like that second movie I showed. And in this case as well, if one samples close enough to the native structure, the lowest energy structures one samples are very close to the experimentally observed structures. This is also true for the other biopolymer that folds in nature, RNA molecules. This shows a low-resolution structure of an RNA molecule folding up. And again, we have the same basic observation. This is now for RNA showing the same types of plots I showed, energy versus RMSD, where the native structures are, in this case, in red at the bottom. They're generally lower than what we can sample starting from an extended chain. Again, on these plots, each point represents the lowest energy lowest energy structure sampled after a low resolution followed by a high resolution search. So one can see that for small RNAs, it's the same overall picture.
Can you say for layman, what do you mean by low resolution?
Okay, so the low resolution.
How do you decide what are the degrees of freedom that you keep?
Yeah, right. It's for in a protein, one has the degrees of freedom along the backbone. The degrees of freedom are just the torsion angles. In the low resolution model, we integrate out all the side chain degrees of freedom. So those are.
When you say integrate out, you freeze them.
Well, no, they're not frozen. We essentially it's essentially like integrating over all possible degrees of freedom for the side chains and taking the mean interactions that they would have with other side chains and with the backbone. Does that answer your question? Yeah. So they're sort of the interactions of the side chains are averaged out to the extent that their precise conformations is not important. All right. So now one thing that you've seen here are these free energy gaps. So the native structures of proteins, the experimentally determined structures of proteins and RNAs are lower in free energy, as shown on those plots, than the ones that we sample when we start with an extended chain. And this is really a recurrent feature of every problem we've looked at of this sort, that we always see a free energy gap. So why is this? First of all, I showed you for soluble proteins. I showed you for multimeric proteins. Well, I think I told you for the protein-protein docking problem, we see the same thing. For membrane proteins, we see the same thing. And for RNAs. And what this is a reflection of is that really the remarkable thing about biological macromolecules is they fold at all. They fold to more or less single unique states with Boltzmann weights of something like 99.9999. You observe only the native structure and closely related structures. Now, this is rather remarkable, of course, given the huge number of states that are accessible to for example, a protein chain as an astronomical number of possible states, yet we only observe one. The only way this is possible is that we must have a very, very large energy gap. The native structure must have a very much lower in energy than any single non-native structure, otherwise we wouldn't observe that structure at equilibrium. And so the prediction problem is really possible because the magnitude of the actual free energy gap is much greater than the error in our ability to calculate free energies. So, of course, we can't calculate energies or free energies very accurately, but because there must be, in actuality, a very, very large gap, we can still, prediction is still possible. And so because of this, the real challenge has been, how does one sample close enough to the native structure, to the experimentally determined structure, to be able to identify it based on the dramatic decrease in energy? Okay, so how do we sample close enough? Because clearly, imagine you're sampling a landscape with a huge valley or deep hole in the middle. If you happen to sample close enough to that hole so that the energy drops, there will be no problem at all detecting what the native structure is. However, if you don't sample near that basin, you will never be able to detect. You have no chance of succeeding. Okay, so of course there are smarter algorithms. One can try and integrate or collect all of the information about the landscape one gathers in these many, many independent searches, which essentially each end up in a different local minimum. We've and of course more computing helps, because the more we can search, the greater chance we have of actually landing in the global minimum. And I'll talk a little bit about using distributed computing for this. One can use experimental information. For example, one can use related structures and start closer to that global minimum, which definitely makes it easier to find. One can use other types of experimental data to hone in on to guide the search, to restrict it to the area around the global minimum. And we've also started to see recently, by developing a computer game called Foldit, whether people have some intuition for where these global minima lie. Okay, so to illustrate the first point, again, imagine that you have some huge landscape. You've landed on some new planet. You're searching for the lowest elevation point. Our approach is essentially you send out many, many different explorers that parachute down onto this landscape and land in different places, and then search around for the lowest elevation point in the vicinity, and then report back what they found. So this type of strategy, where each of those searches corresponds to first a independent low-resolution search followed by high-resolution search. If you have a relatively small amount of fraudsters working on this, say, different explorers, one gets a map like this. Again, this is energy versus RMSD to the true structure. In this case, what we've done is now people running Rosetta at home all around the world get different random number seeds. So they carry out a different low-resolution search followed by a high-resolution search, and they report back the lowest energy structure. And you can see there's a lot more sampling now. Each one of these points is a calculation done on a different person's computer. And in this case, there was one lucky person who effectively won the lottery, who found a much lower energy point than anybody else. And you can see it was close enough to the native structure to have, it's starting to fall into the native basin. In these cases, the structures can be quite similar. When people run this calculation on their computer, they actually see the movies that you saw. And it was after staring out many of these movies that people started writing in and saying that they thought they could do a better job in the coarse-grained part of the search than the computer could. And that's what led to our efforts to make it interactive. And so people can now participate in this search. So we're sort of doing a cognitive psychology experiment to see to what extent can people actually help in this very difficult global optimization problem. All right, so I mentioned that one thing one can do is start closer, and this is a big practical problem in biology. As there are increasing number of structures determined, it becomes increasingly likely that for any sequence, there's already a structure known of the related sequence, and what one can do in that case is simply drape the sequence one is interested in upon the structure of the related protein, and some examples of that are shown here. So in red is the structure of the related protein, and if you just drape your sequence upon that structure, you get the red one here. The actual structure of this protein is shown in blue, and in green is what happens if you start at the red structure and carry out that high-resolution search from there. And what one sees quite frequently is that because you're starting closer, the green is the lowest energy structure one finds in many different searches, starting with the red one, and you move quite significantly towards the actual structure. I think there's a better picture here. This is from a just something, a protein structure challenge that just actually completed a couple of weeks ago. So here's the starting structure of the related protein in red. In blue is the experimentally determined structure, and in green is the lowest energy structure that one obtains in a search starting with the red one. So we can you can sort of locate the lower energy native structure starting with the structure of the homolog. This also works for membrane proteins. This is just another example of the same thing. This is the beta structure of the beta-2 adrenergic receptor was solved. It's in gray here. There's a challenge to predict the structure of the adenosine receptor, which is shown in purple here. And again, starting with doing many independent searches, the lowest energy model is the one shown in pink here, which reproduces some of the structural changes that occur in the core of the protein. Okay, now the problem with everything I've told you about is because this space is so big for any given prediction problem, the chance it's very likely that one won't actually sample close enough to the native structure to find it, and so the methods I've described so far don't have a big aren't extremely useful in biology because one can't be sure that once the predicted structure is correct. However, if you have even a little bit of experimental data to focus the search in the correct region of the landscape, then one can do very much better. And I'll illustrate that now for diffraction data, NMR data, and electron microscopy data. So this is another example from this CAP structure prediction experiment, where here we have the lowest energy structure, predicted structure. Here is the actual structure. They're very similar as shown here. This is like I've shown other slides of this where the predicted structures are very close to the actual structures. In this case, there were diffraction data available for this protein. That's how the crystal structure was solved. But as you know, the diffraction amplitudes aren't enough alone to produce an image of the structure. One needs phase information as well. One needs a phase for each Fourier component in addition to a diffraction amplitude. So this shows what happens if you take that phase information from the predicted structure. So if you Fourier transform the predicted structure to get phases, combine those with experimental diffraction amplitudes, you get a very, very nice map of the molecule. So these models can be useful for obtaining phase information for X-ray crystallography. In NMR, nuclear magnetic resonance experiments, one can get information about the chemical shifts, the resonances of the protons in the molecule, and from those resonances get information about what the local structure is because they're sensitive to the local structure. If you use that information to guide that initial search that I showed, that very first movie, and do everything else in exactly the same way that I described, carry out the low resolution followed by the high resolution search, one finds that one gets structures, lowest energy structures, which are very, very close to the actual structures solved independently. So one of these colors is the independently determined structure. The other one is the structure that comes out from this computational procedure, and they're very, very close. So this method is proving to be a very rapid way of getting quite accurate models from NMR data that's actually quite simple relatively simple to collect. And one can combine this type of NMR data with that symmetric folding I showed. And so this shows an example of an experimentally determined structure and a predicted structure of a dimer using this chemical shift information. So this combination seems to be quite powerful in getting structures quite rapidly. Finally, from electron microscopy, one can get sort of envelopes for where the polypeptide chain runs. And again, starting with chain tracing through this envelope, one can carry out the high-resolution search with all the atomic detail and improve the models quite a bit. Let's see, this is my one last example. This was a paper I got to review quite recently where someone had gotten the Rosetta program and completely independent of us had used it first used it with chemical shifts to generate a model, and then calculated phases from that model and combined that with diffraction data and very quickly solved the structure. So this seems to be becoming something that structural biologists can use quite generally. There's Jamie Williamson and Jack Johnson at Scripps. Okay, so that's an overview of sort of where we are at computation of protein structures, and without experimental data, it's still quite open problem where sampling seems to be the limiting problem. With experimental data, it's starting to look like it can become a practical tool for structure determination. Okay, now we're going to talk about the inverse problem, where now we start with a structure and we want to find the lowest energy sequence for that structure. So now I'm going to show you, now notice we're keeping the backbone structure fixed. What we're changing is the identity of the amino acids that fill out the core of this protein. And this search we can do very, very rapidly. Again, it's the same description of the energy that we're using in the structure prediction calculations. Is that here we're letting the amino acid sequence vary. So the first calculation, of the sort that we did a number of years ago, was to draw on the back of an envelope essentially a protein structure which didn't exist in nature, and then to use this process to find the amino acid sequence which was very low energy in that structure. This was the structure we were trying to make, and after computing the structure and making the protein in the laboratory and experimentally determining its structure, we found that the structure adopted by the sequence that we designed to fold up to this structure actually did fold up to this structure with very high precision. This is a comparison of the crystal structure of the model we were trying to make and the actual structure that was determined experimentally. Moreover, this protein was extremely stable, much more stable, at a much lower free energy folding than naturally occurring proteins, because we had really designed it to have very low energy. But this protein did absolutely nothing. We designed it to fold up to a stable state, and it did that, but it's completely inert biologically. So what we've been really focused on since then is to design new protein functions. So, for example, designing new protein-protein interactions. And this is something that Sorel Fleischmann, who's a student near Bental here, is working on. I'll tell you today about designing enzymes which catalyze novel chemical reactions. We're also working on designing endonucleases that will cut DNA at very specific locations, so we can go in and target sequences, for example, in genes that have been mutated. And finally, working to design HIV vaccine, and the idea here is to design proteins that don't look like something we draw on the back of an envelope, but mimic the HIV surface protein, so as to elicit an immune response to it, to the Achilles heel regions of the virus. What I'm going to focus on today is design of enzymes. So enzymes, of course, are catalysts that catalyze chemical reactions. The way they do that is by stabilizing the transition state for chemical reactions. So to design a new enzyme, after we've chosen a reaction that we want to catalyze, we start by computing the reaction transition state. And then once we have a model of the transition state, we design an ideal active site around the transition state. We position side chain functional groups in positions optimal for catalysis. We maximize transition state binding affinity. And then, so basically, and this part is completely disembodied. We have no protein. We have the transition state with the chemical reaction, and we have functional groups positioned sort of disembodied in space. And then the final step is to design a protein that realizes that active site. And I'll illustrate this with a very simple chemical reaction, the chemilumination reaction. In this reaction, we're breaking this bond here between the nitrogen and the oxygen. You see it's broken here. And this is catalyzed. To catalyze this reaction, we have a base here, which abstracts this proton here, and then we're going to put a positively charged group here to stabilize the developing negative charge. So I'm going to go through the design process for developing a new catalyst. So the first step is to choose choose what the groups are we're going to use to carry out the catalysis. In this case, we have the transition state for the chemical reaction in yellow here, and we're going to use a negatively charged carboxylate group to abstract the proton, a positively charged lysine side chain to stabilize the negative charge. We put a ring up here to promote electron delocalization in the rings here. On this side, in contrast, we've used a histine residue as a base to abstract the proton, with a carboxylate backing up the histine and a serine making a hydrogen bond here. So the basic point is the basic idea here is we place these functional groups around the transition state in positions predicted to be important for catalysis. Once we have those placements, we then search through a large set of protein scaffolds for places where we can realize this active site. That is, place this transition state model and then place side chains such as to realize all these geometric relationships. So this is really a geometry problem which we developed very fast algorithms to solve, and so we search through hundreds of different scaffolds to find the sites where one can realize this site the best. And then the final step, and this isn't a very good picture, is to use the process shown in that design movie where the amino acids were changing to vary all the other amino acids around to bind as tightly as possible to the transition state. And the ending, what one ends up with are illustrated here. Again, the transition state for the chemical reaction is shown in yellow. Here, this is the mechanism that was on the left. We have a negatively charged carboxylate abstracting the proton here. We have the ring on top, and we have there's a hydrophobic pocket to bind the transition state. Over here we have the second mechanism. We have a histine abstracting the proton, and it's backed up by this aspartate here. Again, there's a hydrophobic pocket. So far this is all in the computer. The next step is to, we of course know what the amino acid sequence of this designed protein is because we have a model for it in the computer, and we can make a synthetic gene. Actually, it's very easy now just to purchase from a company a synthetic gene which encodes this artificial protein which doesn't exist in nature. And then once we get that synthetic gene, it's a pretty easy matter to purify the protein from E. coli. And when we do that, we find, when we're lucky, that the designed proteins catalyze the chemical reaction. So this is the amount of product versus time. And that when we mutate the key residues, so this is the one that used the glutamate to abstract the proton, when we mutate it, change that residue, we completely lose activity. Then on this case, this is the one that was on the right that used the histine. When we mutate, this is the design that we were aiming to make. It's active again. If we mutate the histine away, it no longer works. All right, then with the problem is that these designs, while they have activity, are not highly active. There's a lot missing, and to learn what's missing, we're collaborating with Danny Tofik, who's nearby, who has really a lot of experience in using methods more akin to natural evolution to identify mutations which increase the activity of the enzyme. So simply by making large numbers of mutations and screening for those which increase activity, he's managed to increase the activity of this and other designed enzymes by several orders of magnitude. And so in this process, we can learn what's missing from our computational model. Because, of course, what we'd like to be able to do is design from scratch very, very highly active catalysts. So this is another chemical reaction, and I just want to show you a picture of the active site for this is the transition state for that chemical reaction shown in yellow. And in this case, we we've designed in a lysine that makes a covalent bond with the substrate. And interestingly, we found in this case the most effective approach was to actually incorporate an explicit water molecule that's positioned by two hydrogen bonding residues we've designed in to carry out some of the chemistry. And I think that there may be a parallel between our use of our finding that using water molecules to carry out some of the chemistry and the reason why is it's very hard to position many amino acid side chains in exactly the right way. And we think that nature, early in the evolution of enzymes, may have made more use of water molecules that were gradually replaced by more precisely oriented side chains as the systems got tuned by evolution. So these enzymes are also active in a way that's dependent on the catalytic side chains. This is a reaction which breaks a carbon-carbon bond. So there are all sorts of things that one can imagine doing with enzyme design that are kind of fanciful. Here is a scheme for developing a new pathway for fixing carbon dioxide. So carbon dioxide, of course, is a one-carbon unit. One can convert it to formaldehyde and other one-carbon units. And what we've developed now is an enzyme which will take these one-carbon chains and link them together to form 2 and 3 carbon units. In this case, we didn't start from scratch. We took an enzyme which catalyzed a reaction on 2 much larger molecules that was similar in chemistry to benzaldehyde, shown here, and we redesigned the sites, a little bit hard to see, so that it would fit instead a 1-carbon unit. And this enzyme indeed does link these 1-carbon units together. So we're now trying to build up pathways that use this mechanism. All right. So as I said, when people carrying out Rosetta at home calculations watched the screensaver on their computer, they wrote in and said that they could help out. We're very interested in seeing whether both for this global optimization problem in the prediction case, as well as in the design problem, whether people there's a lot of energy out there in the world and people playing games, computer games, and Foldit, which unfortunately I'm not able to demo here because I couldn't get an Ethernet connection. But if you just go to fold.it, you can try it out. It's an effort to let game players all around the world try and work on both these prediction and design problems. And of course, the neat thing about the design problem is that people can make really compelling designs. They can be experimentally tested. And let's see. Oh, this just gives one example of a type of problem where people actually seem to be better than computers. So here's a problem where in the starting structure, here is this hydrophobic side chain that's sticking out, and there's a hole inside. This is a hard problem for, so when the human folded players see this hole, they see this hydrophobic thing sticking out, they simply rotate this piece of backbone around, and the folded player who got the lowest energy, actually we had to take the negative of the energy and multiply it by 100 to get something that looked like a computer game score, but so it was the highest score, actually managed to get the side chain to go in, whereas Rosetta, this high-resolution search, simply doesn't, it's not directed in that sense, so it tended not to be able to do that. So we have some anecdotal evidence that on some classes of problems, people can actually be quite good. All right, so I've talked about the work of a lot of people. Let me acknowledge the people who did this work. For the structure prediction work, the Mike Taika and did the comparative modeling work. Ingemar Andre did the work on the symmetric dimers, and Patrick Barth did the membrane protein structure prediction I described. On the incorporation of experimental data, the NMR data was done by Batson Raman in collaboration with Ad Bax and Yang Shen at the NIH. Riju Das did the work on the combining with diffraction data. The enzyme design work, the Daniela Rothfussberger did the work on the Kemp elimination. Alex Angolini developed the algorithms. Justin came up with and designed the one-carbon sugar converting enzyme. And Foldit is a collaboration with Zoran Popovic and is the work of 2 very talented graduate students, Adrian Choi and Seth Cooper. and I'll take questions there.
Questions, please.
Yeah.
First of all, it's a great talk, and I think for young students, they maybe will start to believe that there is a design evolution. But anyway, you showed us many structures of small domains that are highly packed. Can you use your softwares and these systems to design or to actually predict the structure of multi-domain codings? This is the first thing. And the second question is regarding small domains like what you showed us that actually fall around small molecules like, let's say, PhD domains that wrap peaks or wraps that wrap around GPT.
Yeah, well, let me answer the first question first. So there's a reason I showed you smaller systems. That is because the size of the space that has to be searched grows exponentially with the number of degrees of freedom. And so for larger systems, it's effectively that the search problem is intractable unless we have a lot of information on the region in which we have to search. So if we have no information, we're just given the amino acid sequence, it's like landing on the surface of Jupiter and finding, there's just no hope of finding the global minimum. So the places where we tend to focus on smaller systems because that's where, again, we have a vastly smaller search problem. As far as designing proteins that bind small molecules, yes, that's something that we're quite interested in. The enzyme design is sort of an example of that because we're interacting with a small molecule and then carrying out some chemistry on that. We're also working on methods just for getting tight and strong interactions with small molecules.
Can you do a whole cycle, namely you start with design, go to sequencing, now apply your Rosetta on the sequencing, would you get back
you're absolutely right. So there should be
the same thing?
Yeah. We in fact do that all the time. It's a very important internal consistency check because you could imagine having some structure, designing a sequence that's very low energy in that structure, but then it may have some other structure which is still much lower energy in. So in fact, for most of the
especially you can choose somehow your course kind of degrees of freedom,
Yes.
then they can lead you to some totally different thing.
That's right. No, that's right. So in fact, when we make a design, before we actually experimentally test it, we do exactly the calculation that you've brought up. And if it does something completely different, then then it's clearly not a good design.
Another question,
Yeah.
as a physicist.
Yeah.
I mean, in all of this game, you sort of don't use any kind of physical information about forces or tensions.
Well, yes, it's in
You have some kind of blocks that you fit together, but you don't somehow.
Well, no, that's not quite true. So I didn't talk too much about the way that we're computing energies, but it's quite. So we have, we've got all the atoms are there, including the hydrogens. We're measuring. Really, the dominant force are van der Waals interactions. You essentially have very close packing. The dominant. So in macromolecules, you have no voids. If you have any hydrogen groups that can make hydrogen bonds, they hydrogen bond. So really, the energy is dominated by the extent one can get very, very close packing, while polar groups that are buried make hydrogen bonds. So those details are. Yeah. So. Yeah.
Yes, I was wondering whether when you have those energy gaps at minimum point, if there's a general reason why there is that gap, some physical explanation for the gap, or is it different in each case?
Let's see. No, I think the reason. Okay, physically, what the gap comes from. Let me answer that, and you can ask the next part. Physically, what that comes from again is that it is the very close packing of atoms in the core of the protein. That it's extremely hard to get random. When we fold up a protein from scratch, we tend to get structures which are much more crudely packed. So it's very difficult. And when the structures really do pack very tightly together, it produces a large drop in energy.
Okay, so it's not something you can sort of guess in advance.
Well, it's a general property, but it's different in every case because it's like every jigsaw puzzle is different from every jigsaw puzzle, but they're all perfectly packed.
My related question was whether there are any cases where we have two gaps.
You mean two different states of the protein?
States or metastatic states.
Yeah. Well, you can have two different states in cases where, say, the biology evolution is actually selected for two different stable states, and there are a number of cases where there are a handful of cases where proteins, for example, flu virus, has one stable state, that metastable state when it's on the surface. When it gets inside, there's a conformational change to the actual global minimum. So in those cases, you get your one metastable state and one global minimum. But for most proteins where there's been not selection for such multiple states, it appears there's just one global minimum. Yeah, well, we use that in two ways. One way is I showed that if there's a structure for the homologous, the related sequence, then we can start with that structure. But alternatively, if one has a set of sequences that are all related and you don't know the structures for any of them, you can predict the structure for each independently and then look at the intersection. I think that's that we did. Yeah. Well, it must bind. It either binds the substrate for the reaction more tightly, or it binds the transition state more tightly. The protein itself may not be more stable, but it's interacting more strongly with the transition state for the chemical reaction.
Okay, last question.
Yes, I wanted to ask maybe an naive one. When you're looking at energies, comparing energies, you're calculating energies that, of course, you are something at final temperature. And the question is, how does entropy come into play here, and it's significant in determining this structure?
Yeah, well, okay, there are two large contributions to the entropy in biomolecular systems. The first is the entropy associated with solvation. So when the hydrophobic effect has a large entropy component. That part is included in our calculations. You'll notice I sort of went back and forth between free energy and energy. What we're really computing is the free energy without the configurational entropy contribution. So the
entropy which is related to the folding.
Yeah, so that's the configurational entropy. We're not calculating that, but remember what we're trying to do here is we're comparing single states. So we have a million, we may generate 100,000 or a million configurations, and we're searching for the one that has the lowest free energy. We're assuming that the configurational entropy of all these states is the same to a first approximation, which is reasonable because these are all single discrete states. So the answer is we're including the solvent contribution of the entropy, but not the configurational entropy component. And our assumption is that the configurational entropy of all these single discrete states is roughly the same.
Thank you very much again. I suggest that some of the people sitting on the floor reminded me of a story about an early translation system, computer translation system. There was a story that the Russians and the Americans developed an excellent automatic translation translated from Russian to English and from English to Russian. And then they gave it one sentence to translate both ways. The sentence was, "The flesh is low, but the spirits are high." And the double translation said, "The meat is rotten, but the rock possessed." So I don't know how was the meat, but I'm sure that our next lecture will be excellent. And our next speaker is Professor Martin Grubeler, who is going to ask you a question whether fast folding is physics or evolution.
Let's see if we can keep the spirits high for the rest of the afternoon. The lights, whoever is the expert on. All right, I think. And this is a small enough room, everybody can hear me all the way to the back. I assume. Well, maybe I have to get rid of the floods. He teaches in here, he knows where all the buttons are.
Well,
however, it is somewhat of a search algorithm now underneath. There we are. So I'm going to start by thanking by thanking Tel Aviv University for hosting this and for all take the time to basically work on this award, since I didn't get a chance to do it in the 30 seconds of my first. Thank you. And then I really would like to thank right up front all the people that really do the work, because ideas are really like the sand at the beach next to the hotel where I was in the morning, but to actually make them work and come up with something, it really takes students to do it. And in particular, Pratim Dar Thing, Lee Sung-Jun Kim, Wei Yang, Joby Sabelko, Hubi Nguyen, and Ricky Ballou, whose work I'm going to talk about. And also collaborators, the three groups here in particular for the talk, Jeff Kelly, Klaus Schulten, and the duo of Pitera and Swope at IBM. So I'm going to talk about folding physics or evolution, and fast folding specifically. So, freeze up. Let's see if we can do this one more time. There we go. All right, now it's going to move along. So the first thing I need to explain is how fast is fast. And here we have a time scale that goes from picoseconds to year kind of, or long time scales. And one of the really big differences between folding reactions and sort of organic small molecule reactions is there's a very large discrepancy between the speed of the elementary event, which is on the order of picoseconds for reactions of small organic chemicals, versus the actual time the reaction takes. So I can take some compound like methanol, put it in a bottle, leave it on a shelf for many years, and nothing's really going to happen to it. With oxygen, sitting above it, even though the thermodynamic stable state would be to make some carbon dioxide and water. In the case of proteins, there's a much smaller difference between the time scale of elementary events and actually finally the very fastest folding reactions, which will take place in microseconds, and even maybe what you sort of more typically will find when proteins fold following ribosomal synthesis. The other thing I want to point out is, while a protein may be hard at work for some time, equilibrium constants are not infinitely large. They may be tens of thousands or thousands or thereabouts, but proteins will actually spontaneously unfold and refold again, maybe assisted by chaperones, maybe not. I'll show you some examples at the very end of the talk where the protein will manage to do itself, even in a living cell. But eventually something is going to get to that protein as it shuttles back and forth between the folded and unfolded states. And so, contrasting a protein folding reaction with an organic reaction in terms of a free energy surface, we have really a very narrow reaction coordinate, maybe just breaking of a bond, with a very high barrier, whereas in the case of protein folding, we have a very much broader reaction coordinate that includes collective dynamics of many bonds in the protein, and a much, much smaller barrier, leading to this much smaller dynamic range. So actually, when we're saying that proteins have barriers, we're really already saying that proteins hardly have any barrier at all anyway, because they're on the order of a few kT compared to the hundreds of kT kinds of barriers you can get in bond-breaking chemical reactions. So I say this to sort of maybe make it easier for you to buy in the end that if the barriers are that small already anyway, maybe it's not all that outrageous to have no barrier at all in a folding reaction, because that's one of the things I'm going to talk about. Why would you worry about fast folding proteins? Well, it turns out one of the models that has been developed, the energy landscape model since the mid-90s, to sort of provide a statistical mechanical description for protein folding, makes a number of interesting predictions Some of them are very similar to what already people more or less understood, back 10 years ago. But some of the most interesting predictions come about when you are comparing to systems that have low free energy barriers. And basically, two of these are, first of all, that there is in fact no unique structure-folding pathway relationship. Biochemists used to like to talk about pathways where I go from A to B to C and eventually I end up in the folded state of the protein. And these types of models, and I'll show you some data to support that, basically say that there is not necessarily any unique relationship. So I could have all kinds of ways that something can fold to a specific kind of structure. Now, secondly, that there's fundamentally no physical-chemical reason for barriers during protein folding, but instead all you may end up being left with is a minimal amount of roughness on a free energy landscape. So barriers could even be down on the basically KT level, where just spontaneous thermal fluctuations are in fact sufficient to get you with very high probability over the barrier. So the central paradigm in the energy landscape model is this idea between a correlation between enthalpy and configurational entropy that takes you down into this funnel-like shape where you start with lots of extended configurations at high configurational entropy and a small number of configurations in the folded state here at low enthalpy. And basically, if this kind of surface here is sufficiently smooth, this then automatically produces an energy gap between the ground state of the system and excited states. And that actually is one of the reasons that you can have a unique structure. In a way, actually, the way you can think about it is that this kind of surface here can ensure that your excited states of the system are, that you're basically designing not just for folding, that's one thing you have to do, but that there is, so to speak, a design against misfolding. Not only do you want to make sure that all the interactions are there that make a protein fold efficiently, you also want to make sure that you can't have interactions that would stabilize states that you don't want to have stabilized. Now, from the point of view of doing experiments, though, this kind of plotting really doesn't do me any good at all in our lab. And the reason is that I don't have an entropy button that I can push on any of my experiments anyway. If you do, let me know. So we have to do things the old-fashioned way, and therefore do them in terms of free energies, where you have this enthalpy-entropy compensation. But what this model actually predicts, if I claim that there is really no reason to have significant barriers, is that not only can you go down in energy, but you can actually also go down in free energy, basically by having, because folding is such a complex process that involves all of these many different contacts being formed, that at each one of those little steps, you can compensate some enthalpy with some entropy, and basically never end up having to go over those huge hundreds of kilojoules kind of barriers that you might end up seeing in ordinary chemical reactions. So to give you the sort of bottom line of the talk, right at the beginning, and then hopefully I will provide some supporting evidence, or at least some selection of the supporting evidence. The first point here, no unique structure folding pathway relationship, but we all know that when we do experiments that such pathways do seem to exist. I mean, we do measurements on proteins, they do go from state A to state B. So how do you explain that then? Well, it's actually very simple, right? The Boltzmann factor comes to the rescue, right? If I have two populations in an experiment of two pathways that might lead to the product, then, at least in a sort of quasi-equilibrium thermodynamic sense, the population ratio here is going to be weighted by the free energy difference, and this being an exponential weighting factor means that even if I have an alternative pathway that's 1 or 2 kT only higher up in free energy than the main one that I'm looking at, you already have 90% of the population going through the lower one. If it's 3 kT, then it's, the signal to noise in the experiment is usually 21, 31, maybe 100 to 1 if you're really lucky, doing one of these kinds of kinetic measurements. And so you're not going to see it, even though there may be additional pathways available. Why do barriers exist? Proteins fold pretty fast, but they still usually take milliseconds, seconds, sometimes hours to fold, not the kind of microsecond time scale that I mentioned here. And so, there's probably a number of reasons for it, but the fact that we can make these microsecond folding proteins tells us that it's really not because of some basic problem with the Hamiltonian, the protein couldn't fold that fast. They can, and I'll show you some examples where we've designed proteins to fold, hundreds or thousands or 10,000s of times faster than the natural protein will fold. But nonetheless, the natural protein folds considerably more slowly. And there is a number of reasons, and I'll just mention two of them in the talk. Evolution for function is one, and evolution against protein aggregation is another one. So barriers, it turns out, can be bad for protein aggregation, and it turns out that function is not always compatible with fast folding. As a matter of fact, I won't have time to touch on this anymore in the rest of the talk, But we actually have at these days, basically a nearly 100% hit rate in making a protein fold faster. You give me some protein, and I will make a mutant of it, and I can pretty much guarantee that mutant is going to be more stable and faster folding than the wild type. And what we do is we basically just take bundles of sequences, align them, look for all the residues that are highly conserved. Those are usually functional residues that carry charge or dipole moments on them, or maybe things that allow the protein to flex when it binds to DNA, things like that. And we just take all of these out and replace them by nice rigid things and hydrophobic things, and the protein, voila, it won't function anymore, but it will fold much faster. And we've got X-ray crystal structures of these proteins, and they have the same X-ray crystal structure as the original protein. All you've done is basically just replace the residues that are necessary for the thing to actually do its job, which is really what evolution evolves for, by residues that make it much more stable, make it fold much faster. So then, you could ask questions like, how small are the smallest possible barriers that you really can get? And you'll see that they are indeed very small. And what could you use these things for? Well, I'll just mention one example. You could use them to actually provide feedback, for instance, for molecular dynamics simulations and improve the current generation of force fields, because they're at least fast enough you can actually compare them to extensive MD simulations. Turns out you could also use them as scaffolds for designing new protein functions. All right, so before I launch into a description of the sort of kinetic experiments that we do to understand how we can get this fast folding behavior, even downhill folding, let me talk a little bit about folding as a phase transition, because we actually use thermodynamic tuning to get our answers. That's sort of the key technique in our experiments. It's folding of a 2.5-dimensional object, first of all. If you take any protein out of a database, the PDB, and you put a sphere at its hydrophobic nucleus, and you ask what is the mass of the atoms contained in that sphere as a function of the radius of the sphere, it actually scales as r to the 2.5. And that's because actually proteins, they are kind of a jigsaw puzzle, but one that doesn't really fit all that well. And the reason is that you always have to work with the same 20 pieces you get to cut. In a 1,000-piece puzzle, you have 1,000 pieces and they all have different shapes. Here we only have 20 shapes to play with, and you can't make it doesn't tile things like Penrose tiling or something like that. So you are basically stuck with a small number of building blocks. And what that means is that you have little cracks and crevices and places in this protein, and actually the size and number of these cracks increases in this kind of scaling with the size of the protein. So the bigger I make the protein, the higher is also the chance it will have a bigger crack somewhere in it, because you couldn't fit all the side chains together perfectly. And that's a source of frustration. If I had infinitely many side chains, or if I could have things like half a methyl group or stuff like that, I would be able to eliminate these things and turn proteins essentially into three-dimensional objects. But I don't have half a methyl group. I have to deal either with 0 methyl group or one more methyl group that I can stick onto the system. So looking then at the free energy surface, the way we sort of normally expect it to look like is that there's a basin for the unfolded state, one for the native state. There's going to be a free energy difference between them as a function of some reaction coordinate, which I'm going to leave unspecified for now. At some temperature and pressure, maybe at some different temperature and pressure, the folded state will be the one that's higher in free energy and the protein will be unfolded. And so we can construct a sort of phase diagram for this: temperature, pressure, free energy. And as it turns out, and people suspected this already nearly 40 years ago, the region in which a protein is stable in this phase diagram is more or less elliptical in shape, which means that I can unfold proteins, for instance, both by going to high temperature in this direction here. Here's a plot that shows the secondary structure content by an experimental measurement by circular dichroism and an enzyme phosphoglycerate kinase. So I can unfold it by heating. I can also unfold by cooling. This is a little weirder, because that's kind of like saying, well, if I take liquid water and I cool it down, it'll freeze, and then if I cool it down some more, it would actually melt it. We know that's not going to happen. And again, as David was already alluding to, hydrophobic forces are important. Of course, that involves ordering of the solvent molecules. And this phase transition here, even this one here, you might still be able to get away without talking about the solvent, pretending that configuration entropy is everything. This one down here, you're not going to get away discussing that one without discussing the entropy of the solvent molecules. But then likewise, you can move along in these two directions in the phase diagram. You can do high pressure experiments, and you can actually do negative pressure or tension experiments. And we do some of these also, but let me just mention one of these here as a brief example. So here, what I have is an NMR spectrum. The protons of specifically actually on a histidine residue in ubiquitin, and there is a difference in the chemical shift between the folded and the denatured state. So you can actually tell how much of denatured and how much of folded protein you have. And what we do is an experiment where we just take the NMR tube and we pump with a vacuum pump on the solution so that there's only a bubble of solvent vapor left. and we seal the tube off, heat it up until the bubble has disappeared, and then we cool it back down, and the surface tension actually of the water keeps clinging to the tube, so you get to negative pressures now in your NMR and do this experiment. This is a trick, by the way, it was figured out in the 19th century by Berthelot, but it works well for us too. So, and as a result, what you can see here is that indeed the denatured state is stabilized now compared to the folded state in this NMR difference spectrum, showing that again we have this kind of parabolic behavior in the free energy, both in the temperature direction and in the direction of changing the pressure or applying tension to the system. And these are the kinds of perturbations that we apply. So while we're looking for downhill folding, we're going to tune thermodynamic parameters and look for unusual signatures that are characteristic of energy barriers disappearing from the picture. So basically what we do in these experiments is illustrated here. I visited China a few years ago, so I was at the Great Wall and had a friend of mine take this picture here. And what we basically do to the protein is just this. This is the actual real orientation. What he did basically is to just turn his camera by 30 degrees. I don't have Zen abilities of actually standing like that, obviously. I can barely stand straight here the day after the flights, although I didn't have as bad the airplanes as your connections. So basically, it's just the difference between European and Chinese architecture. They basically, when they build castle-like structures, just put the ramparts parallel to the ground. They don't care, it could be a 45-degree mountain. It's just, and so I had been fooled into thinking, just because you were looking at this background motif here, but in fact, I'm actually barely able to hold on this thing and prevent myself from sliding down. So we're basically going to apply tuning to the protein to force it to go downhill. And the way we do this is by tuning the temperature or pressure, and I'll talk mainly about temperature tuning experiments here. So the idea is you take the protein, you apply a short pulse to it with a heating laser. This heating laser can cause the protein to become more stable because of this cold denaturation effect I talked about, or maybe less stable if you're jumping to very high temperatures. And then you're sending in some sort of probe beam. It could be infrared or even UV light or something else to actually detect the state of foldedness by this particular probe. And actually each of these probes you can think of as a different kind of reaction coordinate. UV might, for instance, report on the local packing environment of a tryptophan, infrared on some hydrogen bonding, so you can pick yourself the reaction coordinate you want to look at. And so we induce the protein then to go from the denatured state to the compact state, or vice versa, depending on how we do this experiment. And again, the idea is, just like in that photo there, to take this free energy surface that may have a barrier and to apply a bias to it. And eventually, if I make the bias big enough, if I already have a small barrier to begin with, I can make it disappear and just have the thing become an unstable system that has only a single well left, basically. So that's the idea behind the experiment. So here it is, just a little bit more quantitatively again. Plot as a function of temperature of the free energy. There are basically two reentrant phase transition points. One here the cold denaturation, here the heat denaturation. So if the free energy is greater than 1, the protein is unstable outside here or below this temperature. And then this is the zone of stability where the free energy here is below 0. And then there's going to be a point of maximal stability. And the hope basically is when you're doing this kind of experiment is that you take a protein from either over here, and you make it more and more stable by lowering the temperature. Maybe you can also help it with some mutations or other things. And so you're taking this free energy surface that looks like this bistable well, and you're just tilting it until eventually things can just slide down. Now, this isn't necessarily always going to work, because you can imagine, for instance, that if I have a protein that's less stable, so here my minimum is not quite as far down as it is over here, it'll still fold, but not quite in a stable state, that as I play this little game and I try to tilt my surface, when I reach the best point, I'm just still not biased enough, and so I still have a barrier, and then I already begin to cold denaturation. So there may be proteins that you just, they're not stable, and you just can't force them to actually do this. Okay, so that's basically the idea. What that means, of course, is that there's going to be a critical temperature of stability of what this melting temperature has to be, where if the protein is less stable than that, you won't be able to get it to fold, basically without a barrier. And if the protein is more stable than that, you will be able to get it to fold without a barrier. We can actually use this thermodynamic model. I'll spare you the math; it's pretty straightforward. But anyway, we can actually use this thermodynamic model to calculate what that temperature is. And so here's a plot of this for a specific protein, the WW domain, which is a triple-stranded beta sheet signaling protein. Here's the melting temperature. Here's the of different mutants, let's say. Here's the temperature at which it would begin to fall downhill if you can tilt it enough to actually go downhill. And as you lower the melting temperature, eventually this curve here becomes vertical. And so there's a certain melting temperature below which you really are not going to see any more of this downhill folding, at least according to this thermodynamic prediction. So what we then did, with this specific prediction here, is to actually make about 30 to 40 mutants of this protein. Some of them were made by some friends of mine at Scripps that actually, instead of doing site-directed mutagenesis, inserted artificial organic molecules as bridges to make turns in the beta sheet. Actually, here's the structure on the next slide. And we just measured all of their folding rates, and what we basically saw is that you get this S-shaped curve where there are some proteins that fold very slowly, that have low melting points, and you get proteins that fold very rapidly, that have high melting points. And then there's sort of a transition region, and indeed, that transition region lies between 50 and 55 degrees Celsius, which is exactly what we predicted by the thermodynamic model, where it should happen, that basically the bistable well ends up disappearing from the picture. The other thing to notice in this plot is that a couple of the data points here at the fast end are actually plotted as these triangles. And what that refers to is a different thing that happens when barriers become small, which is that normally if you have very large barriers, you can't actually observe any of the population at the transition state of the reaction. There just isn't enough of it. But if you imagine lowering the barrier, eventually you will be able to see both populations in the wells, and you will see populations that's diffusing across the transition states. It's sort of, so to speak, like if you can't get to the transition state, make the transition state come to you, by lowering it down. And what that means actually is that instead of getting simple exponential kinetics in this bistable system, you will actually be able to see both the diffusion processes and the activated processes simultaneously. And so what you actually expect is that, if you have a big barrier, you see a nice single exponential kinetics, and when the barrier gets smaller, you start seeing more complicated kinetics. And then when the barrier completely disappears, you might actually see simpler kinetics again, because you now only have the fast diffusion bit. So you can sort of tune between activated at the one end, diffusion at the other end, and then this combination of activated and diffusive folding. And that's exactly what we observe on this side. And the only place where we see this kind of multi-exponential behavior is for those very fast folding mutants that have the high melting points, not down here in this end. So the first time that we actually saw this was in a protein that we couldn't even fold to the native state downhill, but simply to a molten globule back in '99. And again, the idea here is, if I plot as a function of temperature and reaction coordinate by free energy, here's the attractive basin of the folded state, here's the attractive basin of the heat denatured protein, the cold denatured protein, that there's going to be some perfect temperature where I'm as stable as I'm ever going to get between the heat and cold denatured states. And at that temperature, if I'm lucky, I might be able to go downhill, whereas if I heat up the protein, I'm going to go over a barrier, and if I cool down the protein too much again, I am going to go over a barrier. And as it turns out, we actually have quite a bit of anecdotal evidence that it's much easier to take small proteins or proteins that to get them to fold downhill, or to actually just get a protein that might fold through several steps to fold downhill to a molten globule. So if you don't have a hydrophobic core that's all too well packed, it's actually easier to make the reaction go fast than something that has to really click in a very specific way, get water sheets expelled from the core, and basically do all of those sorts of things. One thing I want to mention here is that an important difference between a kinetic experiment where you're at constant temperature and a thermodynamic experiment where you're tuning the temperature and scanning the stability of the protein. That, for instance, on this particular type of free energy surface, if you do thermodynamics, you would never see any evidence of downhill folding. And the reason is simply that by destabilizing the protein, by, so to speak, applying that tilt in reverse, you are actually creating a barrier. So the actual experiment that you're trying to use to detect whether there might be a barrier or not will actually create a barrier in the folding process. And so you are then thermodynamically going over this barrier. So here's another example of this, one more, of a free energy surface that starts out as this double well. We bias it, more downhill, and finally here, if you give it a really good tilt, and experimentally we do this by making mutations that ever increase the stability of the protein, prevent the likelihood of trapping, and as I said, remove functional residues that might slow down the collapse of the hydrophobic core, things like that. And indeed, when you get very unstable proteins, you can fit the data perfectly well to a still single exponential. When you get very, very stable proteins, they fit again to a single exponential, but a much faster one. And in between instability, when you go through this transition where it's not quite downhill and it's not quite activated, you end up getting kinetics that can only fit to this kind of multi-exponential. So that doesn't necessarily mean that downhill folding always has to be thermodynamically hidden. So again here, this example one more time, free energy is a function of reaction coordinate and temperature, where at low temperature I can go downhill, at higher temperature I may have to go over a bimodal barrier. And what you actually expect then from a system like this is that as you do a temperature tuning experiment, even if it falls downhill here at low temperature, that by the time you get to the midpoint of the transition, so you're sitting over here, you basically have a barrier, and so you get this nice, still S-shaped curve, even though the protein may be downhill folding here, downhill unfolding over here, but it will actually fold over a barrier in the middle here. Well, that doesn't mean that we couldn't redesign proteins to lower even this ridge on the free energy surface here. And we and a couple of other research groups have worked on this. I'll show you an example here from our lab, where we took the protein lambda 685, a 5' spiral fragment of a viral DNA binding protein. And here is a version of it, that mutant, that folds in about 30 microseconds. And you can see by measuring by a bunch of different spectroscopic techniques, you essentially get the same answer as far as the stability of the protein as a function of temperature. So there is a well-defined melting point here. But if I make this version here that folds just 3 times faster by making a substitution here at position 33, I find that under exactly the same measurement conditions on the same machine, that we now end up getting a spread of about 10 to 15 degrees in the melting curve. So this particular protein doesn't have a well-defined melting temperature anymore. And the reason is it doesn't have a high enough barrier to actually produce a separated, folded, and denatured state. And therefore, there will be population, as I do my thermodynamic tuning experiment, even in between along the reaction coordinate. And if my infrared or fluorescence or CD and all the various measurements that I do, if they switch at different positions along the reaction coordinate, they are therefore then going to record different melting temperatures. All right, here's a computational example of a very small, just a simple hairpin trypsin tool. And one of the things that we can do also experimentally, and you can see it also comes out that way in the calculations, is to if you reduce the folding barriers by these tricks that I mentioned about, and you get these proteins that end up folding in microseconds, and this one here I'll call a peptide, small enough, that you can actually expose whatever roughness is left on the free energy surface besides the activation barrier. So normally your kinetics are dominant by this one dominant activation barrier. But if we eliminate it, we can actually look under the hood and see what's left over. And you can see there's considerable free energy fluctuations in these surfaces. This implicit solvent model, now they have actually repeated the calculation with explicit solvent, gives very similar answers, predicts fluctuations on the order of about 1 kT. And so what we did is we did an experimental measurement on this, and we've done it with other proteins since then, where we simply change, for instance, the spectral range where we look. So we're looking at a different reporter along the reaction coordinate. And lo and behold, you end up getting different folding times, sometimes a little bit slower, sometimes a little bit faster. And if you measure an ensemble of these times that depend on the detection technique, you can see again this thing folds very quickly in a matter of a couple of microseconds. You can then basically take the rate fluctuations and take the logarithm of those and actually turn them into a free energy fluctuation. And in this particular case, we find that the variance in the free energy is about 0.6 k squared t squared, which, so about 0.8 kt if I take the square root of that. So pretty close to what they actually saw in the simulation as far as the residual ripples that are left over on the free energy surface. And those we think you cannot get rid of, again, for a number of reasons, including the fact that you have a finite amino acid alphabet, and so you can just not have a perfect design that will eliminate any amount of frustration on the way to the protein actually folding into the native state. The other thing that I mentioned at the very beginning is that when protein folding barriers are so small, or you can even tune them to be essentially non-existent, or within the thermal energy basically, another one of the consequences is that indeed you are not going to have a single folding mechanism anymore. And I already showed you that we can make this lambda repressor protein fold two-state, over a barrier, or we can make it fold downhill. And we've redesigned it in various ways to make it fold two-state or three-state or downhill, or to collapse first, but not form secondary structure, or form secondary structure first and then collapse if you like the framework model better. You can pick basically whatever you want. Here's just one example of that, where we redesigned the protein to actually form a folding intermediate that forms in about 50 milliseconds, whereas the collapse of the protein takes on the order of 400 milliseconds. So it takes about 10 times longer. So we can take this protein lambda repressor, this 5' button that I showed you, and I can make it fold by any mechanism that you want, just by redesigning its sequence. So there's clearly no unique mechanism by which this protein has to fold. And it kind of makes sense again, because from an evolutionary point of view, even though you don't want to have, of course, too many low energy states, because then you have a problem of populating these simultaneously, if you have paths with enough of a barrier, there's no harm in having several of these paths have barriers that are not too far apart from one another, allowing you sort of multiple alternatives of getting from product, from the reactant down to your goal or to your product. And the reason this works is because these relatively small free energy differences between different paths, again because of this Boltzmann factor, will quickly give you a large population ratio. And so in any given experiment, it will look to you like the protein has a unique mechanism. But in fact, actually, if I just change a couple of side chains, I can lower the free energy of another path by a few kT, and then I follow that particular mechanism. And of course, if you want to have a protein that can actually evolve, where you can basically change the amino acid sequence without grave consequences like it not being able to fold anymore, this is actually an advantage to have these sort of multiple paths at your disposal, even though maybe in a given realization of the protein, only one of them is getting mostly used. Okay. So if barriers during folding are not obligatory because we can redesign various alpha and beta sheet proteins and so forth to basically fold with very small barriers just on the order of kT, then how come natural proteins do have barriers? Because most natural proteins do not fold in a microsecond. They end up taking considerably longer times, corresponding to barriers that are not enormous, but at least on the order of 5, 10, 15 kT, something like that. And one of the reasons, which at least I saw first discussed in the literature by Hans Schmidt at Bayreuth University, is evolution against aggregation. So the idea is that if I have one of these super stable proteins that can fold downhill, sure, it takes a lot of free energy to get up from the fold state to the denatured state, but it doesn't take a lot of free energy to make smaller excursions. And even smaller excursions that just partly unfold some small piece of the protein can be bad news because these pieces can start sticking to each other now and we can get aggregation events. So in fact, even though I pay the price of making the native state a little bit less stable by putting a hump here in the middle along the reaction coordinate, I now prevent these excursions and that can actually be useful. Now if that's true, what that would say is that these proteins that we designed with great effort, that are so stable and fold so fast and have so little function, that these proteins should aggregate much more easily than the wild-type proteins. And indeed, that's true across the board. So when we take, for instance, wild-type lambda repressor, that's a protein you can put at 4 millimolar, that's a high concentration of proteins, in an NMR tube, and nothing, no precipitation, nothing is going to happen. I go to just 173 millimolar of the fastest folding mutant of this that folds 100 times faster than the wild type, and I start seeing that the folding rate actually becomes concentration dependent, slows down, and the reason for this is that you now get a competition between aggregation and folding of the protein. So these things may fold very efficiently, but that also means they can partly unfold more efficiently, and that's not a good thing. Another reason is evolution for function. Proteins evolve to have a function. This function may not even involve any foldedness at all. We all heard of natively unfolded proteins. But function is often not compatible with efficient folding into a compact structure, mainly again because various of the motifs like binding loops may be entropically unfavorable, or because charged residues may not lead to as fast hydrophobic collapse. There's a number of different reasons. I'm illustrating one here, but this example where I have this WW domain again, triple-stranded beta sheet, and the wild-type loop is a rather long loop, and it's necessary for cognate binding to a phosphorylated peptide. So this is part of the signal transduction function of this protein here. And basically what we did is we made every conceivable modification of this loop that you can imagine, making it shorter. A couple of them are not even shown. We'll put in 6 glycines at one point, to connect the serine with the arginine here. And basically what happens is invariably, as we make this loop shorter or move the active binding residues around, every single mutant that we make folds faster and more efficiently than the wild type, which is the black residue. This is the log of the folding rate as a function of temperature. We have simply not been able to make a mutant that folds, so we worked hard. So we've done everything we could, but we simply cannot get this protein to fold any slower than the naturally evolved protein. Conversely, though, when you make these very fast-folding versions, what you basically find is that the function of the protein disappears by any assay that you do. So here you really have a case where this thing has evolved to have a certain function, and this function is not conducive to efficient folding or forming a thermodynamically very stable state. As a practical application of fast folders, let me just mention one of them, refinement of molecular dynamics force fields. People usually show their successes when they fold proteins by molecular dynamics simulation. I want to show you one of the failures. So this is a case where we redesigned the WW domain to fold in about 10 microseconds, and then we've run now about 25 microseconds actually of trajectory on this. And this is the real structure. And here is what the charm force field folds the protein to. And this is not a trapped state, or we didn't give it long enough. We have actually done enough free energy sampling on this. This state is significantly lower in free energy in the charm force field than the native state of this protein. So charm will fold, but it will not fold to anything that resembles the native structure of this particular protein. On the other hand, if I give charm a heat expander, it will fold beautifully into a heat expander. So you have to pick your force field depending on what structure you want. The hope is, of course, that because we now have this kind of experimental data that allows us to speed up proteins and meet force fields that were halfway in between in the microsecond regime, is that we can also use this kind of data as a further a measure for qualifying and improving force fields. On this one, we won't get anything better than D-. It's folded at least into something, so it didn't give us an F. It gave us a D- on that one. And here's my last slide, just to make a transition to the next talk that we're going to have, which is you would expect that if the folding energy landscape can have barriers that are made that low, that it could be very malleable, and at that point, of course, the environment becomes very important. So, for instance, the presence of a protein in the live cell may be modulated strongly by crowding and just the environment in the cell. And so what we are doing these days is actually experiments where we look at folding in live cells. And so basically it turns out cancer cells are very hardy yeasts, and you can actually take them, stick them in a microscope, express a protein in them that's been labeled so you can tell whether it's folded or unfolded by energy transfer, and then actually take a movie of this with millisecond time resolution, apply a small temperature jump for the entire cell and the water bath around it, and actually change the folding equilibrium of the protein within the cell, and then ask, how does this depend on position in the cell? And it does. There are significant fluctuations in folding rates in the cell, and in this particular case, you can actually see that, so here's where my heating laser is on. I'm unfolding the protein a little bit. It takes about a second here to form this folding intermediate phosphoglycerate kinase in this particular case. And then when I turn it off, I get relaxation back. You can see I'm not quite satisfying like linear response here. There's a little bit of irreversibility because a little bit of ubiquitination or aggregation has occurred. But we can repeat these cycles here quite a number of times and then actually ask questions like, how does the folding dynamics vary from cell to cell? How does it vary within a given cell? And so my time's up, so I'm going to stop here, just leave you with the conclusions that I've already mentioned in the talk, and I'd be happy to answer your questions.
No questions? What determines the time scale of the fast folding? Why microseconds?
Why microseconds? Okay, that's a good question. So the answer is actually, in a way, what the things that you consider to be the reaction core. You could make the argument, and this is a somewhat specious argument, but I'll make it for a moment, that, if I look at the free energy surface or not even a free energy surface anymore, just the energy surface in hundreds of dimensions of everything that can rotate or move in this protein, that it really has enormous amounts of wiggles in it, and somewhere there's going to be one wiggle that's the highest in free energy, and will correspond to a rotation of a single methyl group on some side chain somewhere. Right? And you could say, well, that's the transition state. I know that side chain rotates in 5 picoseconds, so why don't you make it prefactor 5 picoseconds? But really, of course, what we have to ask here is to what kind of extent do we want to coarse grain the problem so that we can describe it in a low-dimensional space. And when you're describing the problem in a low-dimensional space, even in a one-dimensional space, as we attempt here, you are automatically performing this coarse graining. At that point, what becomes your reaction coordinate is a relatively small sequence of events that involve basically diffusion of pieces of secondary structure or their formation in the solvent. And the time for that varies between maybe tens of nanoseconds to hundreds of nanoseconds. And so that's basically what sets that elementary getting time scale here. And there are, of course, sub-events on these and sub-events on those that you can keep going. But those you can't really analyze in terms of a low-dimensional free energy surface. And interestingly enough, though, the averaging is favorable enough that you can get very good results on low-dimensional free energy surfaces. All the ones I showed in this talk are one-dimensional. We've actually shown, and we don't have time to go through it here, that there are quite a few cases though where a one-dimensional surface is not good enough. You actually need higher dimensional surfaces to be able to describe all the observables in the process, and there's no way to reduce it to one reaction coordinate. And this is a general problem with folding or structure formation in the intermediate size system. If I have a small organic molecule, I only need one dimension because I have only one bond that I'm breaking. If I have water and I want to describe its freezing, I have a real order parameter, so to speak, in infinite dimensions, and I again have one order parameter. It's when you have these things that are somewhat halfway in between that you can't necessarily get away with a single order parameter.
More questions?
You mentioned this term soft phase transition. Can you elaborate on this?
Yeah, so why do I dare call it a phase transition at all?
I understood the phase transition, but what is a soft?
Yeah, by soft I simply mean because the diameter of the system is only 20 or 30 nanometers, you cannot get real first order, sharp melting points. They're pretty amazing. They're pretty amazingly sharp, though. The cold denaturation one here is actually even sharper than the heat denaturation one. Over a temperature range of maybe 4 or 5 degrees, you go completely from denature to native state in this particular case. But because of the finite side of the system. I used to not have it in there, and somebody asked me, why do you call it a phase transition? So that's what I couldn't know myself.
I should have mentioned. You mentioned the barriers at least water aggregation. Did you try to check the effectiveness of the proteins that cause diseases?
Well, that's really, of course, an interesting thing to do. We haven't done that yet. So one of the things you might want to do is exactly redesign proteins that are natively unfolded to increase their folding propensity, something like salucine would be a good example, for instance, and then to actually see if you can sort of do the reverse of the experiment that I showed you. Because the only experiment we did was to take things that haptic there and get rid of the there and see that they do aggregate. So you really want to do the reverse also. We haven't.
Thank you very much. Is Jonathan Weissman, I already said Jonathan Weissman, from the University of California, San Francisco. We'll talk about genetics which trial-based probability regulation.
Thank you very much. So, if like me, you had been schooled as a protein biochemist more than 10 years ago or so, you have learned that proteins can exist in one of two conformations. The far more common one are the floppy, highly disordered structures. And then for the unusual sequence that through evolutionary pressure had been selected to adopt a specific globular fold, there'd be a unique or nearly unique structure. And this, as it turns out, has been a really radically, in many ways, poor view of what protein structures can do. Because it turns out that many, and perhaps most under the right condition, proteins will adopt highly ordered beta sheet-rich structures, commonly referred to as amyloid fibrils. And we are just starting to get higher resolution structural information about them, but the common and sort of defining feature, as I said, about an amyloid fibril are their richness in beta sheets. In particular, that you get a cross beta sheet structure in which the strands run perpendicular to the long axis. And what this means is that there's going to be an open-ended phase, and that's critical to these fibrils because what that allows them to do is to adopt or have a self-propagating conformation so that unconverted or non-fibrillar forms of the same protein can bind to the end of these fibers, and that fiber can essentially act as a catalyst to help conversion and growth. A characteristic feature of these fibers are their extreme stability. And there's been a great deal of interest in them lately because the association of these type of aggregates with a wide variety with a number of otherwise seemingly unrelated diseases of protein misfolding. But what's really interested my lab and others in these fibrils is this self-propagating nature and the ability of this self-propagating nature to allow for unique or protein-based inheritance. And my lab in particular has been interested in universal features of this protein or prion-based inheritance, features that are not specific to a particular protein. And so I'll give you a few examples of these and I'll focus on one in particular. So the first, of course, is the fact that they are self-propagated, that they're heritable units. And that, as I alluded to, seems to largely be due to the fact that the fibrils can bind to unconverted protein and promote their conversion. The second feature is that there's often a very high degree of specificity within these, and this leads to phenomena like the species barrier. So, for example, the barrier that was supposed to protect us from getting mad cow disease from cows because of differences between the prion protein in cows versus the prion protein in man. And so, again, this seems to be largely due to the specificity of this growth process, and of course, how these barriers break down is an interesting fundamental and practical question. But the most enigmatic and, in my mind, interesting feature of prion inheritance is that prions come in different flavors or strains. And so apparently the same prion protein can cause very different states or very different diseases, for example, in the case of the mammalian prions. Actually, this phenomena of strains pre-existed the notion of an infectious protein. So back when people thought things like Kuru or Creutzfeldt Jakob disease were caused by slow viruses, virologists were working on this and said, well, if it's a virus, it should have a genome, and that genome should mutate, and that means if we collect samples of this slow virus from different parts of the world and put it back into an animal model, we'd see subtle but heritable differences. And sure enough, there were clearly documented cases of differences in the strains of these spongiform encephalopathies. So differences, for example, in how long they took to cause disease or what parts of the brain they seemed to infect. And these differences seemed to propagate. And that was all fine and good, but then came along the prion hypothesis from Sam Prusner and the notion that what was causing these encephalopathies was an infectious protein, and a protein that was encoded within the individual who was infected. And so now you had a problem, because if genetically identical individuals with presumably encoding the same protein were able to propagate these different strains or variants of the prion, and there was no nucleic acid genome in the infectious unit, what could account for these differences in strains? In essence, you were left with one of two options. Either, the first was sort of, there was something fundamentally wrong with the prion hypothesis, that there was some element, perhaps a nucleic acid, perhaps something else, that we were missing that was key to the encoding these different strains. The second, though, in a way you could argue even more radical hypothesis, is you had to say, not only was there a notion that a protein can misfold into an infectious conformation, but it had to be able to misfold into multiple different infectious conformations, at least one conformation for each of these different strain variants that exist, and these different conformations had to themselves be self-propagating. And so this, in many ways, had sort of been the skeleton in the closet of the prion hypothesis. But it also had been something, I would say, that was, for many years, sort of something that was of interest to the cognoscenti, the limited few who really knew the details of the prion field. But that really changed starting in the mid-90s with the realization that mad cow disease was a different strain or variant of these infectious prion diseases, and that this strain, unlike the scrapie strain that had been infecting sheep and people had been living with and eating sheep and never apparently getting prion diseases for a couple hundred years, that something about the strain, and presumably, if you believed it is encoded in conformation, something about the conformation of the mad cow variant was making it more capable of propagating and infecting people, and a more dangerous strain. And with that, I think a lot more interest came from a practical point of view of prion strains. So, as I alluded to, all of these are really universal features of prions. So many of these phenomena were first documented in mammalian prions, but now we have multiple different prion phenomena in different organisms, and they all, in general, have these phenomena of being infectious, and having species barriers, and being strains. And because of the universal features of prions, it gives us the sort of freedom to study them in the most amenable system. And for us, that has been a rather unusual phenomena in yeast called psi. So first of all, what is psi, and then I'll talk more about psi as a prion. So psi, phenotypically, in terms of is a tendency for some yeast, psi plus yeast, to read through nonsense mutations. So, for example, if you have
AD1 gene. It's an adenine biosynthetic gene with a nonsense mutation, a stop codon in the middle of it. A psi- yeast will stop properly and not produce the AD1 gene product. That turns out to cause the yeast to turn red under rich media and not to grow in the absence of adenine. But psi+ yeast are able to suppress this nonsense mutation, to read through the stop codon, allowing them to be white in rich media and to grow in the absence of adenine. So really quite obvious phenotypes. And of course, there are many different mechanisms of suppressor mutations, the classic of the tRNAs that can read through nonsense mutations. But what made psi so unusual and such a mystery is that, as far as we can tell, to the nucleotide, the DNA in psi- and psi+ yeast is identical. What distinguishes psi- and psi+ yeast is that a specific protein, a translation termination factor called sub35, has altered its conformation from being a soluble functional form, which allows the ribosomes to stop properly, to an aggregated form that's self-propagating. And these self-propagating aggregates do two things. First, because they're self-propagating, it allows the side once formed to be inherited indefinitely. And second, by depleting the cell of this normal translation termination factor, now ribosomes aren't able to properly stop a nonsense mutation like the AD1 mutation that I talked about, allowing through the nonsense read-through phenotype, which manifests itself as this very simple color phenotype. So that's really the beauty of the psi system. You can look at a macroscopic property to streak out yeast on a plate, and just by looking at its color or whether it grows in the presence or absence of adenine, know microscopically what the conformation of this protein is. And I should say one other thing about psi is that the sub35 has a modular domain. So it's got a translation termination factor in the C terminus that's really the globular folded normal protein that we think about that does all its activity. And then on the N terminus, it's a prion domain which mediates this self-propagating aggregate. And in fact, we can take this prion domain of sub35 and put it on other proteins, including, for example, the fluorescent protein GFP, and it will act as a prion. The other really nice thing about the psi system is you can purify this sub35 protein and make it recombinantly in E. coli, purify it to apparent homogeneity, and remove it from denaturing. And after a characteristic lag phase, it will undergo a spontaneous conversion to classic amyloid fibrils, visualized here by atomic force microscopy. Moreover, these fibrils are self-depleting, and that's seen by the fact that you can take a small amount of these fibrils, repeat this reaction where you dilute denatured sub35, remove the denaturing, and now if you have some fibers as a seed, you'll completely eliminate this lag phase, allowing a rapid conversion without delay to the same amyloid structure. But I told you that many, now we think most proteins can form these types of amyloid fibers under these conditions. So the obvious question was, are these fibers really infectious? Are they a prion? And now we were in a unique position to be able to test this. And the technical challenge, though, was how do you develop a system for taking a protein that you produced in a test tube and getting it back in yeast? And, once we formulated this, it was fairly straightforward to come up with different protocols for essentially infecting yeast with this protein. So we first developed a protocol in 2000, then Motomoto and Tanaka and my lab vastly improved this in 2004. And so with this, I was able to take yeast, amyloid fibers of sub35 that were recombinantly produced and creating what we thought might be a synthetic prion in vitro and test the infectivity. What we would see is if we didn't use a protein during this infection reaction, or we used the soluble, non-amyloid form of sub35, and put this back into psi- yeast, you only get small intensity red columns out, showing that you didn't get conversion. But if you use the fibril form of sub35, but importantly, non-amyloid fibrils from other proteins, you get a conversion of fractions of these cells from the red psi- to the psi+, and for all intents and purposes, these converted yeast, these infected yeast, were indistinguishable from the classic psi+. And again, to to nucleotides the DNA had not been changed. You can cure the psi, reinfect, and do this indefinitely. And this reaction can be quite efficient. If you use sufficient quantities in the sort of 10 to micromolar range of the sub-35 fibers, you can essentially all of the yeast converted from psi minus to psi plus. So it's sort of the moral equivalent of Avery's experiment with DNA and showing that that was the effect, that was the genetic material. This showed, I think, at least within the context of the yeast system, that the protein-only hypothesis of prion inheritance, essentially in its most radical form, as first postulated by Stan Pusner, was correct. You clearly had an infectious protein, and it was infectious on the basis of the self-propagating change in conformation. So that, is intellectually satisfying, but in my mind, the nicer thing about this is it gives us a beautiful system for taking advantage fully of all the in vitro work that we could do and the power of yeast genetics and going back and forth between in vitro and in vivo. And that's really what I'm going to focus the rest of my talk on, and focus it on this phenomenon that I talked about of these different strains. The fact that you could have different conformations that were causing different heritable states. So first of all, yeast prions, like mammalian prions, comes in different strains or flavors. In the case of yeast prions, it's actually fairly obvious to see because you have this beautiful color phenotype. So I talked about a red psi minus and a white psi plus, but that turns out to be an oversimplification. Psi comes in different strains that distinguished by the amount of soluble sub-35 and therefore the degree of read-through, which comes to manifest in this color phenotype, and also by the stability by which these different prions will propagate. Once you have a strain, it can be inherited indefinitely through propagation, and you can cure yeast, say a strong psi plus yeast, and is fully capable of forming the full spectrum of different strains. It has no memory once cured of what strain it was there, and there's no memory of once cured because as I talked about with psi minus and psi plus, there's no change in the nucleic acid or DNA of the yeast. So if we were to hypothesize then that really what's different in this is the different conformations of the sub-35 protein, we'd have to, what it suggested was that these sort of simple reactions that we were creating in vitro, where we're getting these amyloid fibrils that appeared, at least at a very gross level, to be quite similar to each other, was hiding a structural heterogeneity. And in particular, structural heterogeneity that could cause these different states. And if we were therefore motivated to look if there really were differences in these different fibers, in particular, if there might be differences, for example, in how the fibers would grow, because that could, in turn, cause these differences in genotypes. So in order to look at this in more detail, we developed, Angela DePesa, from a graduate student in my lab, developed a single fiber assay for monitoring the growth of individual fibers. And the idea behind this assay was quite simple. We took sub-35 seeds, these preformed fibers that had an epitope tag, so that allowed us to monitor the seed, and then drew monomers off of this that would not be recognized by the antibody. Carried out this reaction in the test tube, then quenched the reaction by putting on it onto mica, which allowed you for a platform for a conformational phosphate, added an antibody to now mark, after the reaction had been quenched, the seed, and to therefore follow growth. And this is one of the few assays out there where the cartoon is sort of less clear and less elegant than the actual pictures. You get these, it's in my mind, beautiful pictures where you can see the antibody decorating the seed, and then you can monitor with high precision the growth on the two ends of these fibers. Just to show you that everything is kosher here, if you take a seed that didn't have the tag and grow monomers that do, you get sort of the inverse picture.
What is the scale here?
This would be sort of tens of nanometers. So it's about a 5 nanometer fiber. What you see immediately from these was that there are different types of growth patterns. So there's some that are asymmetric, some that are symmetric, some that are slow growing, some that are fast growing. And these asymmetric, we could completely eliminate the possibility that they were due to some sort of breakage in the fibers by a number of controls. And so this at least was encouraging that there might be really different types of fibers. But if there really were going to account for these discrete strains or behavior of the side, we'd expect that there would be discrete types of fibers. So as a first inkling that this might be the case, what Angela did was to repeat this experiment and simply count the overall growth rate. And what she saw was there wasn't just sort of this mishmash of all different possibilities. Instead, there were clearly discrete states based on how fast they can grow. So we could distinguish, in this case at least, in this one test tube with the same protein polymerizing at least 4 discrete states. Not only that, but once we realized that or could show that there were different states, different fibers with different properties, we were able to develop protocols by repolymerization and changing the conditions of polymerization that would allow us to get relatively pure or very highly pure populations of these different types of fibers. And the simplest, in my mind, the most elegant protocol we had for doing this was to simply change the temperature at which we did the polymerization. The reason why this was nice is you could change the temperature of polymerization, create a different fiber, and then, of course, raise the temperature back to room temperature or lower to room temperature. You have no residue, no risk that whatever you're putting in the mix was causing conformation change. So with this, we were able to get pure populations, say, of fiber 1 and fiber 3, repeat our protein infection experiments. What we saw is instead of getting a mishmash of different types of colors or strains of prions, if we took pure populations of fibers, we would get only pure populations by plus. So for example, fiber 1 gave us a very strong strain, whereas fiber 3 gave us a weaker strain. We could actually purify the prions from these different strains, redo the growth assay or melting the curves, and show that they were in fact propagating the different conformations.
Yeah. What is the colors of
this here versus here?
Yeah. Yes. All in fiber 1. Like the red and the white.
Oh, so the red are just yeast that didn't get infected. But so you didn't have 100% infection rate. So when you do an infection, some yeast don't get prions, but those that do are invariably a strong strain, which meant that all the sub-35 was aggregated and you got a very strong read-through phenotype. Whereas when we took fiber 3, again, some of the strains, some of the yeast weren't getting infected, but those that did get infected were a pure population of strain in which you would get a relatively weak read-through, which meant that you got some soluble sub-35, some compromising sub-35 function. And again, we could change the amount of fibers. One, we used our fiber 3. It would change the efficiency of conversion. So you'd go from a few percent converting to 100% converting, but it would change the strain. So the strains, these differences in the side phenotype were not due to getting multiple seeds versus a single seed. So just to conclude this part of the talk, what we showed is that the infection experiments provide a direct demonstration of protein-only hypothesis and established the amyloids as an infectious or prion conformation, showed that a single protein can spontaneously misfold into multiple different self-propagating conformations, and that prion strains are caused by, or sort of in parlance of genetics, encoded in the different conformations of infectious proteins.
When you said spontaneously, in the language of the first lecture, I again apologize that I'm really a layman. This means that these two configurations correspond to the same energies?
Oh, no, it's all under kinetic control. If you, for example...
It's not like two vacua which are regenerating.
No, they're self-propagating. So once you form one seed, it will dominate over the other one. So if you, for example, at 4 degrees...
some kind of equilibrium?
Once you convert the fiber, once you go to the fiber, it's effectively irreversible on this time scale. So, for example, if you polymerize at 4 degrees, the vast majority of it goes to one conformation. If you polymerize at 37, the vast majority goes to the other. But if you take a seed made at 37 and see the reaction at 4, it remembers the 37 conformation. Because at that point, you don't have a spontaneous fiber formation. You have the growth, you have the conversion dominated by this growth process. So it has a memory of what temperature, the conditions under which the fibers were made. So this was a satisfying result. It provided, an explanation for the strain phenomena. On the one hand, on the other hand, it left an obvious question, which was, why could these different conformations cause these different heritable states. And to give you a sense of why this is a non-trivial question, go to a review from Charles Weissman, two ends and no direct relationships, who of course pioneered mammalian prion work. And in the context of prion infection in the mammalian cell culture system, and he was pondering why it was so hard to get propagation in the cell culture system, he was saying, well, what you'd expect is that the prion would grow exponentially, and then the concentration of prion would divide by a factor of 2 when the cell divides, and it would grow and divide and grow and divide. And if it was growing, if the prion were growing faster than the cells, you'd get exponential growth, and all the protein would be converted. If it was growing less rapidly than the cells, you'd get exponential decay, and the prion would disappear with time. So it's sort of like a fission reaction. You're either gone critical or not gone critical. And it's only in sort of the miracle of this exact balance between the rate of growth of the prion and the rate of growth of the cell that you could have a stable prion state. And of course, it would be a highly unstable equilibrium. But here I'm showing you can get multiple different stable states, each with different degrees of fiber burden, different numbers of fibers. And so, I'm reminded of the famous statement from Albert Einstein that science should be as simple as possible, but no simpler. And so this was too simple a model. And what we really set out to do then is to think about this process, the dynamic process of prion growth with the simplest possible model that could capture some of the true richness of the problem. And for this, we went back thinking about the dynamics of the process, but there was really two different things going on. The first was self-propagating propagation of conformation. So you have a fiber, nuclearly made protein that could then bind to the end of fiber and convert, allowing linear growth. But this, of course, a linear growth in an exponentially growing cell isn't going to propagate. In order to have an effective agent, you have to have a way of breaking these fibers to create new seeds, and this breakage has to, of course, match the rate of, in this case, the doubling of the yeast, so that you can create new seeds for the new cell that divides. We actually know that that unlike this growth phase, which occurs spontaneously inside a cell, this division phase is actually dependent on the host cellular chaperone machinery. This is rather ironic, right? You think of the chaperone machinery as preventing or reversing protein aggregates. But in this case, this aggregated infectious state, prion state, is actually dependent on the host machinery in order to propagate. So we could sort of formalize this with the idea that different conformations of prions could affect either this growth rate or the division rate. And then, again, formalize sort of this dynamic process with a set of relatively simple differential equations that just takes into account how fast the protein is made, how fast the growth occurs in a combination-dependent manner, how fast the division occurs, and then how fast these new seeds get diluted out. In the case of yeast, you'd think the dilution would be occurring just by division of the cell, but in, say, in mammalian prion, we know that the prions are cleared by, for example, proteolytic mechanisms, which would act to dilute out. So whenever you have a set of dynamic equations like this, the sort of critical questions are, how many steady-state solutions are there, and which of these steady-state solutions are stable? So are you at the bottom of the hill or at the top of the hill in the steady-state solution? Turns out that these equations have two steady-state solutions. The first, quite simple one, corresponds to psi minus, in which all the sub-35 is in the soluble form, so your concentration of fibers and amount aggregate of sub-35 is 0. Turns out, though, that when you look at this psi minus state is actually an unstable equilibrium. And what that implies was that as soon as you had a single prion in the cell, you would rapidly be pushed off the top of the hill and propagate down to a prion state with hundreds of potentially different prion seeds. And the nice thing about this is we worked out this equation and an immediate prediction, and that is the unit of infection should be a single prion. And I showed you before, we had already done the experiment of looking at the concentration dependence to the infection. So with this and with the concentration dependence, we went back and quickly fit this to a model in which the infection would require, say, 1 fiber, 2 fibers, 3, or 4 or more. What you can see is all the models that required more than a single fiber for infection had too steep concentration dependence to be consistent with the experimental data, whereas a single fiber infection was very nicely fitted data. Again, arguing that a single seed, once introduced, was able to then propagate and grow to cause full conversion to sunlight state. So the other stable state solution was a much richer one. That was a psi plus, in which a fraction of the protein would be in an aggregated form versus a soluble form. So the degree of translation read-through, this color phenotype I talked about, would then be dependent on the concentration of the soluble protein, which would in turn depend on both these parameters, beta and gamma. That is, the strain-specific parameters on how fast the fibers grow and how fast they're divided by the chaperone machinery. Similarly, the number of fibers in cell would also be dependent in a non-trivial way on these different parameters. We can plot these in a somewhat more intuitive way, in which you would plot the predicted strain phenotypes in terms of how much soluble sub-35 there is, for example, or how many fibers there are, as a function of both the fiber growth rate and the fiber division rate. And what you would get in this is that there's essentially a phase transition where above a certain growth rate and division rate, you could have a stable propagating state in which the nature of that state, or the strain phenotypes, for example, the color, would depend on both of these parameters. Whereas below this, when the combination of growth and division was too slow, you would have a perfectly nice self-propagating amyloid fiber, but it couldn't sustain a prion state. So is this model useful? Is there any experimental basis for this? So we then took advantage of the fact that we could create in the test tube these different conformations of the sub-35 prion and reintroduce them into yeast to explore how these different parameters, how fast the fibers grow, how fast they're divided, affected the overall phenotype. And particularly, we used 4 different amyloid conformations, one called, for sort of historical reasons, SC37 and SC4, which were initially made, and another one called SCS. You can see by a number of different measures, including the melting curves, that they're clearly distinct fibers. We could introduce these through our infection experiment into yeast, and you can see that they cause different phenotypes. So, for example, SC4 was the strongest prion, causing the strongest phenotype and the most aggregated sub-35. SC37 sort of an intermediate level, and SCS the weakest, but all three were clearly distinguished from the psi minus state, and all three could propagate indefinitely. So what were the differences? Why was SC4 giving you a stronger phenotype and SCS the weaker and SC37 in the middle? We went back to our single fiber, this assay, this AFM-based assay, and asked how rapidly was the intrinsic growth rate of these fibers. And what we saw was really quite a surprise. SC4 was the slowest growing of the fibers. The SCS was a little bit faster, and the SC37 was considerably faster. The reason this was a surprise was, I told you SC4 was the strongest phenotype. That was the prion that was causing the largest fraction of the sub-35 to aggregate. This is exactly opposite of what we would predict. Right? You would have expected naively the more rapidly growing fiber would cause more sub-35 to polymerize and therefore cause a stronger phenotype, and that's not what we were seeing. So that's one parameter we're down. What about the second parameter of this strength of how rapidly the fibers would be divided? What we saw here was a much more dramatic difference. This SC4 fiber, so for example, with SC4, SC37, and SCS, made long fibers, subject them to a mild rotation, which causes fibers to break. And we saw this had little effect on the SCS amyloid or the SC37, but on the SC4, it caused them to shatter in many tiny little pieces. This shattering into tiny pieces actually dramatically increased the number of Texas units, so we could take these different preparations before and after rotation and show that the number of prions in the SC4, but not the SC37 or SCS, was increasing with this rotation. We could actually, through really elegant tricks developed by Brian Cox, measure the rate of doubling inside in vivo in a cell and show that this was also true in vivo as well. So the key difference, the key thing that made the SC4 a stronger prion was not the intrinsic rate of growth of the fibers, but the fact that these fibers were much more fragile and therefore able to grow exponentially in number much more rapidly. So back just in terms of this model, what we saw with the SC37 going to SES, the weakest prion, we saw a slower growth rate, but no increase in the fiber division. And that was causing a weaker phenotype in which there's more soluble sub-35 and slightly fewer numbers of prions per cell. Whereas in SC4, you got a slower growth of the fibers, but that was more than compensated by a more rapid division, allowing this to cause a stronger prion state in which more of sub-35 is aggregated, and even more importantly, you get a much larger burden of prions per cell. All right, so that then brings us to the last part of this talk, which is what is the structural basis of the prion strain differences. And so this is, these are sort of, I showed you that there are different conformations and how they affect the physical properties of the amyloid. But the real, $600,000 question here is what is the molecular basis of these different structures? And amyloid fibers are a particularly strong, hard challenge for people doing structural biology because their heterogeneous nature and their large nature makes them generally unamenable to things like protein X-ray crystallography or solution NMR. So there have been some recent progress from a number of labs either trying to get crystals of very small peptides or of doing solid state NMR. But one approach that we've worked on that's been particularly useful is to use the strategy of amide proton exchange. What this takes advantage of is the fact that the amide proton is able to exchange with solution water. So for example, if you take protonated sub-35 and put it in a deuterium solution, this amide will exchange for deuterons. And because the proton can be observed in the NMR, whereas the deuteron is not observed, you can use then the solution NMR techniques as a readout. Of course, it's hard, as I said, these aggregates are very large, many megadaltons, and are directly amenable to solution NMR techniques. But here we can take advantage of the fact that we could do the exchange of the fibers in solution. So in watching which protons are exposed to the solvent and which ones are involved in this phase structure, which would prevent exchange of the proton with the deuteron in solution, spin out the fibers, remove all the solvent, for example, with biooptimization, then dissolve fibers in an anhydrous environment like with DMSO, and now do the NMR on the dissolved fibers in DMSO, which will then give us a history of which of the amides were protected and involved in structure into the solution. And that's really the beauty of this experiment, is that you're able to watch the dissolved fiber under conditions where you get no further exchange, and then from this experiment get a history of what the structure of the fiber was while it was in its amyloid conformation. We were able to get quite nice spectra from this in DMSL. So here's an example of the glycine region. We could count 21 of the 23 different glycines. The non-glycine region for each of these is an amide proton from a different amino acid. It was a little messier, but they were clearly distinct, nice peaks. So we were strongly motivated to try to put names on these peaks so that we could figure out which of these amides were unstructured and which ones were structured in solution, as evidenced by which ones were able to exchange with solvent. The technical challenge is that sub-35 is sort of an intermediate size for the solution in NMR, about 253 amino acids, or 28.5 kilodaltons. But the real problem is a very low sequence diversity. 61% of the end domain, this prion domain, was either glutamine, asparagine, or tyrosine. And then to make matters worse, it was highly repetitive. So there's 5.5 imperfect repeats in the N-terminal prion domain. And the real challenge here is you're looking at sub-35 in DMSO when it's completely unstructured. So you don't get the structure of the fibers as a way of differentiating the conformation and giving you different peaks in different resonances in NMR. But fortunately, really, we wonderful collaborators and help on NMR approaches, and really the NMR techniques have become dramatically improved over the last few years. Still, it was really a rather heroic effort from a graduate student in my lab, Brandon Toyoma. He had to do 20 different 3D experiments that ended up being about 6 months total of NMR time. Made 10 different specifically labeled NMR samples, either things like tyrosine, asparagine, glutamine, lysine, phenylalanine. And with this, he was able to get assignments for 161 and 200 possible residues. So he did 161 different probes speckled throughout the protein of where the structure was. And as some of the primary data shows here, we saw dramatic differences in the exchange and therefore the conformation in different regions of sub-35 in a strain-dependent manner. So for example, glycine 31 is strongly protected. You get little exchange after a minute or even a week of exchange in both this SC4 and SC37 conformation, whereas glycine 51 was also strongly protected in the SC37 conformation, so after a week it was still there, but was fully exchanged within about a minute in the SC4 strain. And putting this together with the really extensive mutagenesis and other approaches, what Brandon was able to see was that these conformational differences were leading to a dramatic increase in the structure of the sub-35. The regions of sub-35 that were in amyloid beta sheet group structure, in the SC4 or SC37, they shared a common beta sheet core at the extreme end terminus. But in the SC37, this core had almost doubled in terms of the number of residues, encompassing another 35 amino acids or so. So that this region was the one that we saw strongly protected in this conformation, but not in this conformation. So, as I said, there's a dramatic expansion in SC37 that increases the stability in the fluid potential chaperone binding sites. And together, this increased stability and the decreased ability of the chaperones to bind and to promote the division of this SC37 conformation, in turn accounts for the weaker stripe side phenotype of SC37. So just to sort of summarize what I've shown you is that a single protein can spontaneously misfold into dramatically different infectious conformations, in particular involving large-scale reordering of the backbone. And really, I think more succinctly, that the rules of protein folding and misfolding are different. And I think one of the reasons that protein biochemists originally found the notion of different strains being encoded in different prion conformations is because we, as David alluded to, have been so trained to think about a folded conformation as a unique or nearly unique thing, an unusual feature of a polypeptide. But that's generally true for globular folds that we think about for most functional proteins. Whereas that's rather than being the exception, the rule is that proteins seem to be able to adopt many different amyloid-type conformations. And second, the strain-specific changes in structure in turn dramatically alter the physical properties of prion. For example, increased structure leads to reduced breakability or frangibility of the fiber, and then this decreased frangibility leads to weaker physiological impact as fewer new prion seeds are generated during infection. And so finally, just leave with you with a hint that maybe this might be a more universal feature and a nice starting point for thinking about the physiologic impact of prions or protein aggregates in mass. And this was a part of a wonderful collaboration or conversation that we've had with Stan Krusner's lab. So Stan Krusner's lab over the years has been studying in mice this strain phenomena in enormous detail. And through the years, he had collected many different strains that were causing different types of diseases in mice and had different incubation periods. And then doing melting curves in guanidine on these different strains in vitro. And in essence, a huge amount of data that it was very hard to make sense of. But with this, in sort of joint meeting that we had, a wonderful postdoc then, Giuseppe Legnami, in Stan's lab, went back and took their old data and then did a simple plot of the quantity and melting point of their different strains in vitro and the incubation times in mice. And what we saw was that there was a beautiful straight line where the stronger fibers, the more stable fibers, had the longer incubation times, and the less stable ones had the less incubation time. I think this gives us a hint too of maybe why it's been so hard previously to create an infectious form of the mammalian prion. Because of course, if you're creating an infectious prion form, what naively you think you'd want to have the most beautiful fibers, the most stable fibers, and those would be the ones that would most resist being diluted or degraded inside the cell, inside an animal. But in fact, it may be exactly the wrong thing, because it's the fibers that are highly stable that form relatively inert large aggregates, whereas it's less stable ones that are able to break and propagate and grow exponentially that could be far more dangerous. And conversely, it gives you a bit of pause when you think about making anti-aggregation or anti-amyloid agents, that maybe an agent that causes imperfections and allows fibers to break more rapidly could actually have a reverse effect and allow them to propagate more robustly. And perhaps an alternate strategy would be to stabilize, sort of paradoxically, stabilize the fibers so that they should be in a relatively neutral large aggregated form. So with that bit of speculation, I'll end and thank the people who really did all this work. Brandon Toyama, a wonderful graduate student in my lab, with two great NMR collaborators at UCSF, John Gross and Mark Kelly, did all the NMR studies. Helmut Speer did the original protein detections, and then Motomosa Tanaka really turned this into a wonderful practical tool and did all the strain work with Sean Collins, who did a lot of the modeling work with Moto. Thank you, and I'll take questions.
It seems like according to this model, you expect that fibrils grown under conditions where sort of destabilized conditions would be the less virulent, whereas ones going under strongly stabilized conditions. Is that what you see?
Yes, so that's exactly right. So the SC4, the 4-degree fibers, are the ones that are more fragile and have a bigger impact. We've actually seen this with a couple of different yeast prions, or you can, as you do at 4 degrees without a little bit of quantity, you end up getting the more stable fibers that have a weaker impact on the cell.
So we use European diseases just by having super stable prion former?
Yeah, that could. So I would say a small molecule that stabilizes it may be.
Or you just give people a seed that just makes mega stable.
Yeah, that sort of depletes the pool. Yeah. And you can see, so the answer is I don't know whether you actually turn that into a practical thing. But what you can see is that if you make yeast with one strain versus another, you can see a competition between the strains that can allow that can essentially pull off the pool from one form to a different form.
So from a evolutionary point of view, saying that stable or long and stable fibers are advantageous or protective? That's what you suggest.
Yes, and I think that I should say I'm not doing justice to a great deal of work from the sort of non-prion amyloid fields like Alzheimer's and Parkinson's, and I think independently and really very different means that's becoming more and more the feeling that it's soluble oligomers that are the most toxic and dangerous forms, and that there's nothing intrinsically cytotoxic necessarily about an amyloid fibril, and that it actually, the fibrils and sort of these agrosomes that you see in the cell may be sort of the cell's garbage can. I should say also, just as an aside to remind, it's an interesting different point, is that Cy is not toxic to yeast. It's actually a highly conserved system, and we think, so there's nothing intrinsically toxic to even a rapidly growing organism like yeast. Yeah.
So the chaperones, if they would break down with these aggregates, which make more harm, and how do you control this kind of thing?
Yeah, I mean, that's, as I said, that's sort of the paradox, is that this...
Do they have a specificity for space in this?
Yeah, so we're very interested in trying to understand how chaperones actually bind to and mediate the breakage of these aggregates. I say one of the key chaperones that's dependent on this is a AAA protein called ATP104. And this is a very good, it's a major thermotolerance gene, CRV or extreme thermotolerance. But ironically, or perhaps not ironically, when you think about this, it's not present in metazoan cytosol. And it may have been that, although it's wonderful at taking apart protein aggregates and rescuing them from aggregates, it has the unfortunate side effect of allowing propagation or replication of amyloid. That said, whether other chaperone systems are able to break apart, there seems to be in the mammalian cells other chaperone systems that can do a similar sort of thing.
The model of transportation depends on the chaperones that break into aggregates and inheritance of the transprotein by cell division. So how do you explain the rapid and widespread propagation of plants in the mammal brain?
Yeah. So first of all, the basic dynamics I think are very similar with dilution occurring by clearance of the fibers through, for example, proteolysis versus division. But mathematically, it would come out in a very similar form. That was one of the surprises that's come from the mammalian prions. It was initially thought it was so stable, you put it in an animal's brain that would last a very long time. But Brusner and other people did radioactive labeling experiments and saw that it was actually very dynamic, that the infectious form could be cleared within 24 hours in an animal. So I think you still have to have this sort of dynamic process where you're continually generating new prions that's competing with the dilution.
How does it transmit from one cell to another?
Yeah, so that's obviously a very important, a critical question, because that would be a critical feature in these types of dynamic models. And if you could intervene with the efficiency of going from one cell to another, that might be a way of sort of going from the critical to non-critical state where it could be cleared by natural clearance mechanisms. So I guess the short answer is, that the exact mechanisms are really remain to be seen. More similarly, the mechanism for division of the prions will remain to be seen.
One last question. So what actually causes the toxicity of our...
All right, so that's a huge question in the field, obviously. There are a lot of different models. I should say sub-35 seems to be an evolutionary sort of mechanism for regulating the activity of in this epigenetic way, but not to be toxic. So for all the wonderful things that these prions can do, one of the things that, at least in this system, is not particularly good at doing is studying the mechanism of neurodegeneration. But it seems the neurons are specifically sensitive to protein aggregates, and a wide variety of neurodegenerative diseases, including non-infectious ones like Alzheimer's and Parkinson's, are intimately associated with these protein aggregations. There are a number of hypotheses from pulling out essential factors to causing holes in membranes to inhibiting the proteasome that have been implicated. And my hunch is at this point there's not just a single simple universal mechanism, because I think if there were, we'd know it. There are lots of mechanisms that do, things that do occur, but proving that they're really central to the pathology has been very hard. And in a sense, until we can sort of reverse it and show that we stop the disease, it's going to be hard to know for sure we have the right answer.
So let's thank again.